Alright, let's make sure the microphone is working. Seems like it is. My voice is a little gravelly tonight, but I think I'm mostly back in normal operating conditions. I'll try not to cough into the, microphone or whatever. I was playing around with this trigger earlier, and I thought I fixed this issue, but it's still doing it, so this is what I need to do. Make sure that I save it. Hey Sarah, how's it going? That's not the current view of the moon. No, that's just a picture that I took. I think it's maybe from 2 months ago. I went out and took a picture, and then I don't know if I streamed that particular view of the moon. You're being hypnotized. Yeah, the spinning actinopticus is a little hypnotizing. How's everybody doing? My my weekend was mostly spent trying to get over a cold, and now it's past. But hey, Pack, how's it going? Just a little shout out for Pacific Plankton. Yeah, so I spent most of it's kind of raspy, but I actually like it when it's kind of gravelly. That's my favorite form of my voice. You get some sort of extra layer of ASMR from me, I guess, tonight, as I sound like vocal fry the whole time, unfortunately. I think the moon is currently only about a quarter full, so it would be the opposite of what's up there on the screen because it's 1/4 waxing. You're going for a run? Oh, okay. She's running on the beach. I think she'll be okay. Yeah, it's a waxing crescent right now because we just went past the new moon relatively recently. We had a clear sky, and I thought I might go out and I was like, oh, it looks really clear. And I went outside and I was like, oh no, it's a new moon. There's nothing for me to stream. So, yeah. Watch out for. There's all kinds of things that might get you on a beach. A horde of angry crabs. I don't know. It could be anything, frankly. Small kids. You got to look out for them. Who knows what they would have laid as a trap for passerbys. So, this sample that I'm looking at is. I'm planning on sending some stuff to science streams. Bloop, bloop. This guy here, Balint, and his wife Lita. And I thought it would be a good idea if I looked around in the sample a little bit and made sure that the samples that I'm sending him are reasonable. Because I made new slides and I didn't bother to look at them yet. And I thought, I'll just take a look at them and maybe see how they turned out and see what's in them. This particular sample that we're looking at is from Marl Lake, Wisconsin. And at some point this past summer, we did some field work. My wife and I did some field work, and we went through Iowa and we looped back and stopped at Morrow Lake in Wisconsin. This is the lake that my wife's also a diatomist, and this is the lake she's currently counting. And because the vials where the samples that were in them were just on the counter, I went ahead and made my own slide of her samples. Just one. But there's a lot of material in there. She won't miss it. And she's already made slides from that sample, so it's not a big deal. But I thought, well, I could just use one of these. This might be an okay thing to send that they wouldn't have seen. And I don't spend a lot of time looking at the Marl Lake material. Originally, it was a project that was started by a master's student in my lab, but the project is, it's one that Carlin is the principal investigator for, my wife, and so she was sort of directing the, I was directing the student, but she was directing the research. So the student eventually decided that they couldn't finish the project, and now she's counting it as a result, which sometimes happens. We have to pick up the projects that our students can't finish because the grant doesn't care whether the student does it or a PI does it. It just needs the work done, and so she's working on counting that, and it's sort of a companion lake to the one that Addison, my master's student who graduated, who graduates this semester, technically, but did her defense in December, so she's been out of the lab and has a job someplace. She counted the Clear Lake stuff, which was the other lake that we visited in the summer, and it's sort of the companion lake to those two. They're part of the same research project. This thing that we're looking at in the microscope right now is tabellaria and a tabellaria, not tabularia. But there's a bunch of little diatoms that are all around it. So let me see. I would have thought ahead. I would have pulled out the little arrow. You get to hang out with one of my grad students tomorrow. You're being appreciated. I'm trying to think of who you're talking about. One of my grad students? Or do you mean just one from our department? Because my current graduate student is in Cincinnati right now at a conference, Laura, and she's the only one that I'm currently advising, unless you mean Addie. Turned me fiery. So this is tabellaria. This little guy over here is an amphora. I actually don't know what species that is, but it looks like an Ariensis or pediculus. This is where did they move it? I think it's Samathidium. No, Samathidium rosenstockii. And this is a Pseudostar Syra brevistriata. This is a piece, just a fragment of a Stephanodiscus. That's a girdle band. These little guys are Lindavia michiganiana. This is a little phytolith fragment. This is a girdle band of a diatom right here. And this is also a girdle band of a diatom. This is a girdle band of the Stephanodiscus. That's the fragment for this thing. And that is a girdle band that matches this one. That is very likely the girdle band of Lindavia intermedia, would be my guess. And this is probably the girdle band that goes along with this little Lindavia michiganiana right here. Oh, Kathy. Okay, yeah, good. Kathy deserves some attention. She's a great student. All right, let's move around a little bit in the slide and see what else we can find. Here's some more. This is Cyclotella michiganiana, Lindavia michiganiana. Let's see, I'm going to put some of these in here, and then we'll try to look at them a little bit more closely. Keep in mind, I literally haven't looked at this slide before. So, hey Evan, how is it going? How are things? We're just looking at some, these are fixed slides, by the way. These diatoms are dead. And these are old samples, so the diatoms that we're looking at are actually I don't know how deep this is. About 77, let's see, that would be about 77 centimeters into the core. So these are probably from the Little Ice Age. I mean, they're fossil diatoms. And this one is Lindavia michiganiana. And girdle bands don't travel too far on the slides. Well, the girdle bands are, they probably were attached to, so, they go between the two valves. And it probably was attached to something in the sample. And they probably either snap off or fall off when we prep the samples, or maybe while we're processing them. So in some cases they're still attached, and in some cases the girdle bands are completely detached, and you'll just find the girdle band out there floating by itself. But these are, as I mentioned, they're fixed, which means that they are hard mounted with a type of epoxy that's called Nafrax, and it has a high refractive index. What that means is that if you were to look at these same diatoms in water, you wouldn't see nearly as much detail as you can with this, because it basically modifies the interaction, the interaction of the light as it's passing through. So it's like it goes through the lenses, and then it hits the oil, and then it gets into this mounting media. And it's designed to make this siliceous diatom skeleton basically stand out a little bit more. And so part of the reason these look sort of spectacular in our field of view is that my microscope is nice, and part of the reason is also that they're mounted in a special media. So you're getting settled into your new house. Oh, that's great. It's always kind of an interesting thing moving to someplace new. Yeah, so this mounting media started off as something that, in the ancient days, somebody built it using basically some kind of airplane glue or something. But they had all kinds of different resins and materials that they tried to try to figure out what had the best refractive index with silica to create this sort of cleaner view. And then they mixed them up in a bathtub, and they would just sell them in small batches to diatomists, because there weren't that many diatomists, and there still aren't. But now it's sort of, there's a website you can buy them from. It's a little bit easier than trying to get somebody to make it up in a bathroom for you. So I want to talk a little bit about this michiganiana. Hey, Samsung. I sound a little different. Yeah, it's not my microphone, it's just my throat. But I'm gravelly. I'm vocal fry because I've been coughing, I guess, probably is part of it. But it's not completely clear from congestion. But I feel fine, and I felt fine yesterday, actually. I just didn't want to stream because I was sniffling, and like constantly, and then periodically I just would have really strong coughs. And today I cough a little infrequently, so I figured I could probably manage it. So yeah, I figured people partly are listening to me, for the way I sound, and if my sound is sniffles and coughs, they're not going to be very happy. So, Dangling, how are you doing? So this surface that you can see, like this half of the diatom is actually in focus, and this half of the diatom up here is completely out of focus. And oh, there I go. Now I'm bouncing. And the reason for that is basically because the diatom has different elevations as you move across the surface. So if we were to draw a straight line through this, and you could look at the differences in the elevation, that line would be shaped like an S. And we call that tangential undulation on the valve face. It's a dip, or this side's low elevation, this side's a high elevation. So you can think of it as basically like a waveform moving across that surface. And as a result, only part of it will be focus at any given time. So I can make the bottom part go into focus down here, and then the top part's out of focus, or I can make the top part in focus, but I can't make them both in focus at the same time. And you can see that this part of the diatom around the edge, so this is called the central area, and this is the striated area, and these lines are called stria. That's just a science way of saying stripes. So it sounds like I'm saying something really cool, but I'm just saying this is the striped area, and this part's in the middle. I think you guys could probably figure that out. But these hard round dots that you see right here and right here, and you can see they kind of line up on this edge. They are central photoportula, so those are a type of opening that goes through the valve, and they stand out because it puts all of them on the side that's elevated, basically, and the other side doesn't have any. And that's the normal pattern for a lot of things that have tangential undulation, is that they basically have this sort of a pattern. So kind of cool. You're walking your dog. It may appear in the sky differently. We can see the moon at the same time. Oh yeah, of course. Maybe you like the sound of somebody else's sniffling. Well, I mean, there's probably people that don't mind it, but I know I wouldn't like it. So I thought maybe other people probably wouldn't like it. That's just how I am. I guess because I only think about myself as my context for how things go. I think, well, I really wouldn't like somebody sniffling the whole time when they were trying to talk to me about science. That's repulsive. So I just decided it wasn't that big of a deal. I could miss a stream or two. Nobody's gonna mind. I just do it for fun. So if it wasn't gonna be fun, we weren't gonna do it. This is another diatom right here. It's actually a set of diatoms. This is two halves of a diatom, but they don't make a whole diatom when you put them together. They make two halves of sisters or siblings. We're attached in a colony together, and this is the colony still being connected, but the rest of the diatom is basically broken off. And this is Oligocyra. This is the end of one valve, and this is the start of the next. And these little sort of nubby teeth that you see right here are actually the bases of little tiny spines, and the spines interfinger with each other like knuckles, and they hold themselves together across the interface between the two valves. So it's the set of spines on this side that matches the set of spines on this side. And the part where they connect to each other, so if you wanted to learn a little bit about Oligocyra, that's this genus. Oligocyra is this guy. And this is Oligocyra ambigua that we're looking at. That's the species and the genus. And oh, paleontologizing is raiding. Oh, thank you. How's it going, Danny? Welcome in. You made my screen explode with a bunch of crazy emotes, and I love it. How's everybody doing? It's been a while since I did a stream where I was on late at night and could have been raided by Danny. So thank you for bringing your viewers in and remembering that I exist. It's great to see you here, all of you monstrous number of people that you brought in. I hope your stream went well, and I'm gonna just take a stab at it and say you were talking about dinosaurs and maybe some really cool discussions about the ecology of dinosaurs, which if you don't know paleontologizing, that's what they do. And really the only place you can go on Twitch to have somebody talk about dinosaurs with you. So if you grew up and like I did, and like my daughter, and you know the dinosaurs better than the adults that are around you, and you seem fixated on them, and you never let go of that fixation, Danny's the place where you should go hang out. He's on pretty much every night, and he does a variety of really cool things. So I think you should definitely give him a look-see or a follow if you're just in here hanging out with me. I don't know what you mean, Protech. Protech? What does that mean? Yeah, you're busting in on my microscopes. Alright. Yes, and thank you for giving Danny a shout out. Every weekday, yeah, great. And I was on a stream the other night and he was talking about he's getting some exclusive dinosaur game where you know, access to it early, and he's going to share that with his streamers. So if you're also into gaming, he also does a little bit of gaming, especially if it relates to dinosaurs. So bear with me a little bit. My throat's still sore. I had a cold over the weekend, and I spent all week basically sniffling and coughing. And I'm at the point now where I'm, you get over the illness, and then you just have some of the symptoms, but you don't really have, like, you're not tired or achy or anything. This is, I guess if I were trying to do a radio voice, it would probably be very effective, but I'm not whispering. This is my normal voice. It's just all fried out because of the coughing. So, but if you'd like me to talk about Dusty's Muffin, or what are some of the other ones that they did on that NPR skits on Saturday Night Live, I can give it a shot. It would be better if I had a panel of people here and we could talk about, recipes with some sort of double entendre. So it would be real. Yeah, sweaty balls, exactly. Sarah knows where I'm coming from. The sweaty balls, the dusty muffin, there's a lot of different recipes that they talk about. Right, exactly. You're dialed in to 99.3, the diatom. Thank you for giving that a shake. Claire, something special happened and you didn't even realize it, which is in all the colors you can change my little magic bubble that I live in here. You can't change it to purple. The only way to make it turn purple is to shake it and then get lucky, because it goes through all of the colors randomly, and then it lands on one, and it can land on purple, and you did it. You made it turn purple. I can make it turn purple, but viewers can only make it turn other colors, and then when you shake it, it's the only way to make it turn purple. So you've got it back to purple, which is its magical color, and nobody can make it. So you'll probably make Claire very angry if you change it, but I can still change it back. All right, we've been looking at just one diatom for quite a while here, which is Oligocyra ambigua, and I was talking about this sample is from a lake in Wisconsin. This is from Morrow Lake, which is a lake that my wife is currently enumerating. She's doing the analysis on it. She's a diatomist as well. And this isn't one of her slides. I made my own slide, but I took it from one of her samples, and she doesn't know about it. So, if you have enough channel points to tell what your diatom spirit guide is, it will pop up and it will tell you. And then it should be attached to you forever. So you are a Solophora. And that's the way you'll be in the channel for all of time. Every time you hit that button, it'll come back to Solophora. And you know what? Let's go find a Solophora, because I saw one in here. We'll just do that in Huggins' name. I'm sure there's some in here. Oh, wait, there's one actually. This is a Solophora. There you go, right in the middle. That is your diatom spirit guide right there. And it's guiding you, apparently, to push the right set of buttons, because there's a lot of them in there that I don't have in this sample, but I definitely did have this one. This is the diatom Solophora pupula, and it's a very common diatom in North America. Slophora pupilla is identified by, it has sort of like a slight undulating side. It bows out a little bit, and then it kind of comes back in a little bit, and then the ends are really, really subtly capitate. And Slophora often have this sort of you see this dark area right here? That is it's like a little silica platform, a thickening, and it creates this very specific dark pattern that we see right there. And that's a common characteristic of Slophora. She's enumerating, it's Marl Lake that she's enumerating. Let's go try to find something really different. So there's a lot of diatoms on these slides, of course. And when we count diatom slides, normally what we would do, let me zoom all the way out. So everything that you see in the field of view, we would have to enumerate and track. What are we looking at? And the reason that we do that is because diatoms tell us something about the ecosystem. Clara, you're Mastagloia. Very nice. One of my favorite diatom genera, by the way. And if Pacific Plankton is still here, she would tell you that the name Mastagloia, you won't believe this, but it's absolutely true. Mastagloia translates from Latin as nipple glue. I don't know why they called it that, but that's what the name means. So, but they're very cool diatoms. Not my fault. I didn't name it. I'm just telling you that's what it means, and I'm pretty sure that Pacific Plankton will back me up on that, because she points out on her stream every time she sees one. So we would have to basically look at our sample, and then we would have to enumerate everything in it. And what I mean by that is we would have to tell what do we see and what species are they. You're tempted to find your diatom spirit, but you like to remain diatom fluid. Well, you're free to choose or not to choose. Nipple tooth, see? See? There you go. That is exactly what I'm talking about. If it starts off masto, you know they were talking about nipples. So, there's a lot of weird names in diatom stuff, and I think it's because the German guys that originally did a bunch of the naming of things, I think maybe they had some really repressed sexual anxiety or something, because the little holes that are on diatoms are called areolae, and the split that runs down the middle of the diatom, this thing, is called a raphe, and these are body terms, and they have a process that's called like labiate process. I mean, these guys were really, really like,
hey, Maui,
I feel like they had some repressed feelings. So yeah, exactly. Maybe they weren't breastfed. It's very, very possible. The thing is, in science, the terminology and its origin don't really necessarily have anything to do with what we call it anymore. I mean, people don't walk around calling them the mastodon's nipple teeth, but you just learn what the word is, and then move forward, right? You only have so much time. Joshua Sweet, thank you for subscribing with Prime. I should point out to people, if this is your first time in the channel, I don't always stream from the light microscope. On Wednesdays, I usually stream from a scanning electron microscope. A little less of the bells and whistles happen because it's a little bit more serious when I'm on the scanning electron microscope. At home, my computer can handle a lot more than the computer that my SEM is hooked up to. It's busy running a scanning electron microscope. So there's a little more fun things that go on here. And I like to let people just play around with whatever, so I hope you're having fun with it. Boba, hello. Let's see, I should do like a little shout out for Boba. There's one for Boba. Boba Binks is an artist, and he draws pictures of birds, and then he converts them into these sort of templates that he creates and carves out, and then uses those to paint. And he paints really sort of intricate and abstract backgrounds, and then puts the birds and cats and whatever else on the foreground of them. And does it all right on Twitch so you can see it happening, which is pretty cool. So for people who also haven't been here before, I also stream photography, birds, the moon, nature stuff basically. Sometimes we look at live specimens. Today we're looking at mounted slides. So sometimes we look at living things that are also like, water bears and rotifers and organisms that you'd find in lakes, paramecium and amoeba. So there's a wide range of really cool organisms, and I'm sort of a naturalist about things that aren't diatoms that I like to stream about. So, I like to keep it educational and try to keep it fun. So, this is so I just wanted to give you kind of an example of what I meant. No, that's not a fake all-charged battery. That would have been a great gimmick, though, Claire. I have a dummy battery in my camera now, and it's plugged in. So my battery will always be on full while I'm streaming. There's no more flashing battery sign. So it'll always just say full because it's plugged in. But I just want to give you an example of what... oh, wait, I wasn't finished. I got a stream for a subscription from Joshua Sweet a second ago, and I wanted to point out that I have a job. I'm a professor at a university, and I don't need Twitch to make money. But I do appreciate when people subscribe to my channel. The reason for that is all of the money that comes in from bits, cheers, subscriptions. I sell merchandise from the Redbubble store with my own artwork and products that are just on Redbubble. I make calendars with some of the diatom artwork, whatever. I don't take a penny of that home. All of the money that comes in goes straight to the lab, and I use it to pay for either supplies that we use during the streams or to help me pay for student research. So if students want to look at something, undergrads that are working in my lab, graduate students that are working in my lab, if they need money for something, I take it out of that money. And so I looked at it the other day. I was trying to figure out how much money has been brought in to support student research in my lab. And I started streaming on Twitch maybe a year and a half, almost 2 years ago, maybe a year and a half or something like that ago. And I've managed to bring in something like $1,400 in that time. That's gone directly into supporting student research in my lab. And I also wanted to point out that right now I have two students that are thank you for the hype train, by the way. And also Sam, thanks for giving a subscription to Claire. I have students in my lab right now that are presenting at the Geological Society of America meeting from the North Central section in Cincinnati, and tomorrow they have posters that they're presenting. And my wife is also driving back and forth between Cincinnati and here. It's about a 3-hour drive because she wanted to be home to read bedtime stories to my daughter, and so she's been down there with my students, and I actually have some of my former students that are currently professors now. They started off as undergraduates in my lab, and they came all the way through my lab as master's students, went on someplace else to get their PhD, and now are professors in other universities. And I have one of my former students who's there that's watching over she calls them my little ducklings, my little students that are there because I can't be there because I'm busy teaching. And while my wife is presenting some of the research from Clear Lake while she's there, I'm watching our daughter, so I'm sort of taking care of the house, which normally my wife does most of the work for that, and I usually am spending most of my time teaching. But so they're there taking care of my students, but I have students that are doing presentations tomorrow. So if you're going to go to the GSA meeting in Cincinnati, you can check them out. I think part of it is also hybrid, so if you wanted to just attend virtually, I think you can. All right, let's look around a little bit more. And then I wanted to give you also an example of, just a really quick example of counting. because I told you that we had to enumerate stuff. I can usually tell something about a sample just by looking at the diatoms and then using something about their ecology to figure out what they represent. And any particular field of view is not going to be super informative. In fact, we usually have to count somewhere between 300 and 500 diatoms for every slide. We have to identify each one to species. And then we look at the transition from one slide to the next slide to the next slide. And we try to tell what's happened from one slide to the next with respect to which diatoms are present. And we use this to basically reconstruct things like water chemistry, the salinity, the amount of nutrients, the depth, the light level. And if you're thinking that's a lot of things coming from a few species, what I can tell you is that diatoms are extremely sensitive to environmental conditions. And diatom fossils are one of the cornerstones of reconstructing paleoclimate for the last 4 million, 10 million years. They're one of these tools that we use for relatively young stuff compared to the kind of things that Danny looks at. But this is how we do it. And one of the things that's really cool about diatoms is because they're so small, and because they preserve so well, and because they're extremely responsive to the types of environments that they're in, you don't have to take very much sample, which means you can have very, very high resolution information coming from that material. It's a small possible fossil, so I'm using a paperclip. It's actually smaller than a paperclip. But just to give you an example of that model that I was trying to talk about, let's see. Hey, there's a Mastigloia here as well. This thing here, Lindavia michiganiana. Here's another one, Lindavia michiganiana. This is Pseudostarosira brevistriata. This thing right here. I'm trying to think of where they put that thing. Detenta is the species name. I don't remember where they moved the genus. Lindavia michiganiana. Lindavia michiganiana, that's a microsphere. That's a particle that we put in the sample so that we can figure out the concentration differences. There's another microsphere there. That's Asteria formosa, but it's just the girdle band, so we normally wouldn't count it. That is the girdle band of this thing. This is Denticula kutsingiana. This thing over here is Geisleria adaptata. This thing down here is a Amphora pediculus, or Inariensis, I'm not sure which one. These aren't my samples, but there are 9,000 known species of diatoms in North America, and I don't know all of them. So, yeah, why can you see through them? That's a good question, Zorro. The answer to that question is these are the skeletons of diatoms, and diatoms are a type of microscopic algae with a opaline silica cell wall. And when the organism is alive, chloroplasts would be inside the cell wall, which is where that structure is. And the fossils that we're looking at just have the opaline silica left, so they're basically little glass, valves or containers that the cell material used to be in. And we've processed them to get rid of the organic matter that was left as debris in the sample. So all you're seeing is these little glass structures that are basically the cell wall or the skeleton of the organism that used to live there. So that's why you can see through them. The other reason you can see through them is this. So that's the light source. It shines up from the bottom of the stage, and then it comes into the sample, and then into the lens, and then up through the lens into the headpiece where you're looking. That's actually the camera up there. And then there's a split where you can see, this is where I can see out of here. And this is what we call a transmitted light microscope. And what that means is the light is transmitted through the sample. And so what you're seeing is basically an X-ray, essentially, of the organism itself. And if you wanted to see what the actual organism looks like, we need a scanning electron microscope to do that. And it happens that I have a scanning electron microscope, but it's not in my house, which is where we are. It's in my lab back at school, so at the university. Okay, so hopefully that answers that question. We're looking through them. Yes is the answer. It looks a little bit like the floaters you've seen. It's possible. So is this a fossilized microscopic organism? Sarah B. Music, it definitely is. This is a type of algae, and the diatoms that we're looking at on the screen, so this thing here is about 20 microns in length. These ones are about 10 or 15 microns in length. That's about 5 microns. This is a microsphere. They should all be the same size because they were produced by humans. And to give you some sense of how big things are, there are 1,000 microns in a millimeter, and then, you know, like 1,000 millimeters in a meter. So if you're thinking about things in terms of, you know, like most people don't work in metric very well, but the general model for that is, you know, like just look at a ruler, and the tiniest measurement on there, on any modern ruler that has both metric and inches, so the littlest tick mark that you can find on there will be millimeters, and it will take 1,000 microns in order to make up one of those little ticks. So this tiny little microsphere, which is 5 microns, there would have to be 200 of these in order for you to get 1 millimeter. That's how small these things are. We're looking at them at 1,000 times magnification, and then my camera's actually got a little bit of zoom on it. The question that Danny asked is, are most diatoms genera monospecific, like dinosaurs? And the answer is no. Very few of them are actually monotypic. The majority of them have more than one species. Some of them only have a few. But the ones in North America that I was talking about usually have hundreds of species. An interesting fact, I actually told my Lakes and Wetlands class today, we were talking a little bit about vertebrates. And, you know, like I threw out this number, 9,000 diatom species. And if you're not used to thinking about the numbers of species of things, that probably doesn't sound very impressive to you. But the most abundant species of vertebrates, just going to give you something that you're used to thinking about, on planet Earth are fish. There are approximately 24,000 species of fish on planet Earth, and if you took all of the rest of the vertebrates on planet Earth and you put them into one bin that was vertebrate but not fish, there would still be more fish species than there are vertebrates. So there are more fish species on planet Earth at 24,000 than there are every other vertebrate combined. So just to give you some sense of things that you're thinking about, you know, like every animal that you see with skeleton, there's more fish. And part of the reason for that is that most of the vertebrates are derived from fish. Fish are tetrapods. Most of the things that we think of as vertebrates, I think, are tetrapods. But just to give you some concept of that, in North America, in North America, there are more than 9,000 diatom species, and on planet Earth, known species of diatoms there's more of those than there are of fish. So there are more diatom species than there are all vertebrate species that you've ever heard of, you know, on planet Earth. Does that count tetrapods as fish in that? No. I mean, yes. Tetrapods, fish are tetrapods. Yeah, right. Now, I'm not saying all tetrapods are fish. I'm saying all fish are tetrapods. So, just to give you some concept, right? So, there's a fairly large number of diatoms in North America. One of the things I am always been impressed with when I go to Danny's streams and he's talking and he shows all these dinosaurs. Hang on a second. This is for Claire if you're still here. That's a Mastigloia. It's just a fragment of one. I think that's Mastigloia grevellii, but I'm not positive about the ID. one of the things I was always impressed with when I go to Dana's channel is that he throws up a picture of a dinosaur and he knows all of them. I mean, he just knows all of the dinosaurs. There's only like, I don't know, like 400 species of dinosaurs, though. I mean, there's not that many of them compared to diatoms. So if you showed me 400 diatom species, I definitely could nail at least 400 of them. So, I mean, not to make it any less impressive, because that's way more diverse than, that's just, diatoms are kind of small, and we have to learn a lot of them. But I'm always impressed by that. I always thought, wow, he knows all of them. You just like point him any dinosaur, he immediately knows what it is. So it's pretty cool. Anyway, let's look around a little bit more in this slide, and I'm going to see if I can find anything that's, there's some really pretty diatoms in here. Oh, here's a really big one. Let's stop and take a look at this. Is a Cimbella. Oh, you have 2,000. Well, I mean, I don't know how things get split, so yeah. Well, a quarter of those are probably not. Yeah, I don't know. I don't know anything about the validity. I went looking for how many diatom species are there, or dinosaur species are there, and I was like, oh, it's only like 400. I always thought maybe there was more, but I mean, if you know that many, it's still pretty impressive. Here we go. This is a Cimbella. That's the genus. That's, hang on a second. We can take any diatom genus from a freshwater diatom. And I can type this little thing, and then a window pops up, and it actually takes you to the web page for the guide from Diatoms of North America. Is right there. Your guide species will only show up for a couple of seconds, and then it's the same diatom guide species for you always, so you don't have to worry about that. You're working on a publication very early stage about dinosaur tax account. Oh. So diatom speciation, if you're wondering like how many are on planet Earth, the answer is that we don't know. If you take all of the fossil species and all of the modern species, I think there's something like 40,000 described species, but there's or more than hundreds of species that are described every year for diatoms. So those numbers are rapidly changing, and I'm responsible for some of that. I've described a new genus and several new species, and I have a paper right now that's close to being accepted. I just need to make some revisions where we describe 5 new species of diatoms. Just to give you like a sense of it, right? And the expectation is that our estimates of species are off by quite a bit. Most taxonomists think that the number of diatom species on planet Earth, when the ultimate tally is put together, will be somewhere in the neighborhood of between 1 and 2 million species of diatoms. And I don't know how valid that is. It's a number that if you go to the Diatoms of North America webpage, that's the one that they actually cite. And so I suspect that those guys aren't messing around. Those are mostly taxonomists that put that page together, and I think those estimates are probably pretty close. So that's a lot of species, and I can attest to this by saying that like no matter where I go, if I take a sample and I start poking around in it, I find stuff that I cannot identify. And it's not because I don't have the skills to identify them or the books to identify them. It's that they probably don't have names. And in my library of slides in my lab, I have dozens of things which I just have marked as SP1, SP2, SP3. They're too uncommon for me to bother to even put a name on them. I'd need to have a pretty large population of them to do a good job describing them, but a lot of it's just outside of the realm of what I have time to do. So anyway, this is a nice example of a Cimbella, and if you're wondering like where did it get its name, I feel like you should be able to guess. We all know what a cymbal is, right? Like the kind of thing that the drummer plays in the band, right? The tap, tap, tap with the cymbals, or like bang together in a marching band. And this has the shape of a cymbal. So it actually has a name similar to the thing that we would think of. Alt Miss Frizzle. Hello, Alt Miss Frizzle. And also, thank you for joining in relatively early and being so excited about diatoms, Miss Frizzle. I saw you here really early and seemed pretty excited when I said we were going to be looking at diatoms for mounted slides today. Your question is, do you think asexual reproduction contributes to or reduces diatom speciation? Don't some of them reproduce sexually? The answer to that question is asexual reproduction does not contribute in any way to diatom speciation. It cannot. And the reason for that is because diatoms that are reproducing asexually are clones. And by definition, clones don't have genetic variability. They are, if they're made perfectly, exactly the same as the thing that sprung them. So they don't have any any way of generating any kind of speciation. However, I want to point out that, so your second part of your question is, don't some of them reproduce sexually? And the answer is all of them reproduce sexually. It's just that diatoms undergo a two-staged sexual cycle. Most of the time, they are reproducing asexually. So they just clone themselves, and they just keep cloning themselves and cloning themselves. And the way that they do that is something that we usually just call binary fission. In other words, we have one organism, and it grows two halves of itself inside of the existing skeleton, inside the cell wall. One half goes with this side of the valve, and the other side grows another half that closes itself off. And then we have two organisms that are just kind of attached to each other, that are used to be one and now there's two, and then they just split apart. And so you end up with an organism that is just cloned itself by basically splitting in half and forming two new organisms where there used to be one. And so this is a kind of confusing concept for people, unless you're used to like thinking about an amoeba, right? Amoeba do the same thing. They just keep constantly splitting themselves. And so an amoeba is effectively an infinite organism. And diatoms, in many ways, are kind of infinite organisms because they just, where there was one, there's two, and there's no way to tell which one was the adult because now you just have two daughters. And those two daughters make two daughters, and those two daughters make two daughters. But when they split enough, because they're creating the valve inside of an existing valve, it causes one of the valves, the daughters will be slightly smaller because they had to make a valve that fit inside of a small valve, right? And then when this one splits, it needs to make a valve that fits inside of that one when it splits off. And when that half then becomes a parent, it has to create one that fits inside of that one. And so the population gets smaller and smaller and smaller and smaller, and they reach some point where it triggers them to switch to sexual reproduction. And there's a huge value to this because cloning yourself is the best way to take over an environment. You can very rapidly replicate, and you can basically colonize anything you want. Which is why diatoms are part of the reason why diatoms are such sensitive ecological indicators. Because if they're doing well, they just replicate like crazy, asexually, just cloning themselves. They're in their perfect environment. They'll just keep making as many of themselves as they can. And then when they get a little bit smaller, what happens is they switch to sexual reproduction because they get too small, right? They get to the point where they're basically unable to keep making smaller and smaller valves, and it triggers them to go to sexual reproduction. And usually there has to be an additional environmental cue that is telling them that they're under optimal conditions. And then what happens is some of the diatoms will basically split apart, and then when they split apart, gametes will come out of some of them. They'll turn into boys, if you like, and some of them will just become eggs that will get fertilized. And so that's the way that, for the most part, that's the way that the centric diatoms, or the ones that are radially symmetric, that's how they behave. And they are the ones that are usually plankton, and they're living in the water column, and they're reproducing like crazy. A typical diatom, if you gave it all of the nutrients that it could possibly need, can replicate under the ideal conditions and temperatures maybe 8 times in one night. So that's not one diatom splitting 8 times. The first one splits. Each one of those splits. So now we have 2 that each split into 4, which each split into 4, right? Which split, so you can do the math. That turns one organism into like 128 organisms overnight. And they can just keep doing that under ideal conditions. And then they can switch to sexual reproduction whenever they need to. So the speciation of diatoms happens during sexual reproduction because that's the only time you have genetic exchange from one species, from one specimen to another specimen. The rest of the time is cloning, and there's no way to have speciation occur when you're cloning yourself. At best, you might mutate yourself if something goes horribly wrong and you can't copy your genetic information correctly. But for the most part, as I was talking about one evening with Ivo Lazi, typically what happens when you get mutation, like point mutations or something, diatoms have photo repair mechanisms. They try to repair their DNA, and most algae do as well. And a lot of the smaller organisms, or organisms that are exposed to UV light, actually have photo repair mechanisms in place that basically say, my DNA is damaged, I need to fix my DNA. And they'll try to repair the damage that's been done by light energy. And so it's very, very difficult. The cloning process produces very stable, consistent offspring, or they get damaged so badly that basically they can no longer survive. So it usually is a counterproductive thing to get point mutations for most diatom species, I would guess, and any kind of algae. But when they turn into the sexual reproduction, then you get genetic exchange, and then you can actually get some speciation that occurs. I want to follow that comment up because this is actually a really interesting conversation. I look at diatoms through time, and I'm a paleontologist, as much as Danny would tell you. I look at evolution in diatoms, and I specialize in looking at diatoms through very long periods of time. I come at diatom paleoecology from the side of a geologist. I'm a geologist by training, which is why I am always in Danny's channel making snarky comments about paleontology, because I'm a paleontologist and I know all the things that I need to know to be a paleontologist. But I function in a realm of ecologists and biologists most of the time, and they're used to thinking on very small timescales, and I'm used to thinking in these very long timescales because I'm trained as a geologist. And so I'm looking at samples that go through and span through millions of years, and I look at some samples and I can actually detect evolution in diatoms from the top of a core to the bottom of a core, or from the bottom of a core to the top of a core, as we would say as geologists. We start at the bottom in time and move towards the present. But I've observed and documented evolution in a number of species in the span of thousands of years. And one of the things is diatoms are very, very capable during those sexual reproduction events of exchanging a lot of DNA, and they appear to be one of these organisms that can very rapidly evolve. And there's many examples, not just the ones that I have documented, but my advisor and some of her colleagues, they have examples of this from Yellowstone Lake, where they had one species and basically they can just watch it grow into 2 different species in the lake system itself. You can actually see it evolve if you look through the sediments pretty quickly in spans of typically somewhere between 2 and 4 or 2 and 5,000 years, somewhere in that range. We can definitely see diatoms evolving. And if you ask most of the taxonomists about how long most of the genera, the diatom genera that we observe, how old are they? I think most of them will tell you that the diatom genera that we observe, there's more than 200 genera of diatoms in the freshwater realm. I don't know how many are in the marine realm because I don't study marine diatoms. But I think they will tell you that most of those things are young, very young genera. There's a bunch of very old ones that are still around, but most of the genera that we've observed have speciated, and they actually suggest that in some cases that it may be human beings that are leading to their speciation, that we are moving them around because we move around, and we keep introducing them into environments that they may not have been in before, and they are rapidly colonizing these places and basically evolving into those spaces, and that many of the genera that we see appear to be fairly short-lived. I mean, in terms of how long they've been around. They're young. And even though diatoms have been around since the age of the dinosaurs, there's fairly good evidence that there were long lineages of pretty stable genera, and then more recently we've seen them just radiating like crazy. And people have made an argument that it's human beings that are sort of creating that environment for them. Okay, so let's see. I found out this rock was a shell nodule after breaking off a bit with your trusty paperclip. Okay, let's see. Thank you for explaining this. I've never had anyone give a thorough explanation. They just say they reproduce viruses by binary fission. I can actually provide you with some pretty good reference material for what to, I think, are you a teacher or something? But I can give you some examples, and there's some pretty good pictures that we often use to sort of showcase that behavior. So they don't teach you about this when you specialize in ruminant nutrition. Well, you can always learn, and I'm glad that there's people here who are interested in this sort of stuff. I've got a lot of knowledge is sitting around in my head that I tell my students, and the ones that are in my lab are excited, but the rest of them don't care. So, especially with those sudden switches to sexual reproduction, diatoms might really typify punctuated equilibrium. So, the idea of punctuated equilibrium always came across to me, Danny, as just a measure of scale. If you look at things closely enough, evolution always happens fairly quickly, and if you, especially on geologic timescales, right? So if we're looking at just a few thousand years and they're evolving very rapidly, I think that it's sort of a matter of scale. If you're taking a long view, and I think also the geologic record is biased towards creating a view of punctuated equilibrium because we don't have all of the data. It's like a puzzle piece. It's like a puzzle with some of the pieces missing. And so it looks like there's this big jump that happens all of a sudden quite frequently. So yeah, you're a 9th grade biology teacher. Yeah, oh, great. I usually like to try to keep my my streams in the family friendly category. So I try to keep people from saying things that would be challenging in case people want to use any of my VODs or whatever as teaching material. And I archive almost everything that when I remember to do it on my YouTube channel, which I never tell anybody about, but there's a YouTube channel out there. If you just Google Diatoms Attack, and all the SEM stuff and most of the light microscope stuff and all the birding and whatever else that whenever I remember to do it, get archived on there. So feel free to share any of that with your classes. I'm happy. I'm here trying to educate people, so I'm happy if people can use it for those purposes. All right, so let's look around a little bit more. I've been staring at that one diatom for a very long time, and it's a little bit easier to do when there's moving things on the screen, but it's hard to talk and run a microscope and sometimes at the same time. This is a Stephanodiscus, a very large Stephanodiscus, and just to give you some idea of how we can use them for environmental information, this very large Stephanodiscus usually indicates elevated nutrient conditions. So if I saw a lot of these, it would suggest to us that the environment is becoming more nutrient enriched, usually from people that are putting some extra nutrients in the environment, but not always. This thing that's back here, as I mentioned before, is Lindavia michiganiana. This guy right here has a transverse undulation on the valve face. It's a little smaller in terms of its actual size. Usually indicates stratified lake conditions. And this thing back here is a fragment, a bit of a dissolved version of Lindavia intermedia. And these things, you're wondering to yourself, well, if this is a lake that's well mixed and has lots of nutrients, and this is a stratified lake, which usually has low nutrients, and this one is sort of an indication of deep lake mixing, or slightly stratified conditions, if you want to think of it the other way. How are they in the sample together? A couple of things. Our samples represent in time. This sample probably actually spans something like 15 years. So what happens in lake systems is in the spring a lake will mix, and when it mixes, basically there's a lot of nutrients available in the water column, and this thing will bloom. And as we move into the late spring and early summer, the lake will start to stratify a little bit, and then this thing will bloom. And then when you move into the late summer condition, the lake becomes very strongly stratified, and then this thing will bloom. And in our sample, we actually have all three of them together. These are the dominant centric diatoms in these samples because they're representing the different seasons. But if I see a bunch more of these, I know that the lake's season may have extended. So we have more spring mixing, or we have more summer stratification, or a longer sort of late spring extension. And so we can look at these to tell us something about how the environment changes through time by looking at the concentration of this one versus this one versus this one in the water column. This process of having organisms that would normally be competing for resources that are living in the same lake system, and we know that because they're collected from the same sediments, that would normally be fighting for resources, but they've divided the seasons up. This one does well in the spring, this one does well late spring, early summer, this one does better in the summer. This is a process that ecologists call nutrient or partitioning, niche partitioning in environment. And it's a way for many organisms to live in the same ecosystem but not have to fight for resources. And so this niche partitioning approach is it's used by lots of organisms. It's used by algae especially, and the diatoms will use this as a way so that this one and this one don't have to fight with each other for the resources that they need. They have different nutrient preferences. This one likes high nutrient, this one likes low nutrient, and this likes something in between. And they have different silica and phosphorus and nitrogen preferences. And they use that to basically occupy that same deep water lake environment where they live, but they can occupy it in different times of the year. And so this is actually a really cool strategy. It's used by many organisms. It's used by by warblers. So if you're a birder, I'm a birder. I know that some of you are birders, but you're only interested in those really old birds, Jurassic birds. But if you're a birder and you know anything about warblers, you know that warblers will also do a similar strategy. One will live in the bottom parts of the same types of pine trees. Some of them will, or they'll hunt for food in the bottom parts of pine trees. And another species basically will just occupy the top parts of pine trees, so that they don't have to compete with each other for those same resources. They just scavenge the part that they are most capable of getting to easily. So yeah, niche partitioning is a pretty awesome concept. You're starting to find this with dinosaurs, and I would imagine that that all organisms have this as a tactic, as a way for them to basically access the same resources and not have to compete for those resources, because interspecies competition is probably the most damaging for them. And so if they can find a way to avoid that, I mean, being eaten is also problematic, but you can overcome that by just producing a lot of offspring or trying to make defensive structures for yourself. But if the resources become scarce, that basically destroys your whole population. So that's a critical concern. I also spotted something while I was blabbing, and that is, usually I'm just looking at diatoms, but that is not a diatom. This is diatom. These are all diatoms. This is Pseudostar Syra brevistrata. That's Stephanodiscus nigeri. This is Lindavia michiganiana. As I mentioned, those are microspheres. And this thing is not a diatom. It's an imposter, but it's closely related. This is a chrysophyte scale. And chrysophytes are a type of golden brown algae, so they're cousins to the diatoms. And they create these sort of scaled body structures. And during the active part of their life stage, they have scales on the outside of their cell walls. And when they go inactive, they create something called a stomatocyst, which has also got a silica component to it. And I can probably find you a chrysophyte stomatocyst because they're often also captured in the same diatom samples that we look at. So we have to be pretty much aware of what all of the little particles that we come across are, instead of just diatoms. And not everything that you see is a diatom, although most of it is in these samples. But there are things that are closely related that are in them. So this chrysophytes stomatocysts are one. Another that we could possibly encounter in our sample is a phytolith, and that is a piece of plant that basically just incidentally usually puts a little bit of silica in the cell spaces or within the cells of the plants. And so the shape of a phytolith mimics a cell wall of a plant cell, and those can be somewhat symmetrical, and so they might confuse you into thinking that they're something like a diatom. There's a nice dense view of some diatoms right there for you. Almost everything in the field of view there is a diatom, except for a little bit of dirt and some microspheres. But hang on, I wanted to just show this while I'm here and we're talking about it. Newt Newt! Thank you for the newts. This little guy right here is a chrysophyte stomatocyst. They look like little Christmas tree ornaments when you look at them in the scanning electron microscope. In the light microscope, let's just zoom in like crazy people. Hey, Crowl and Fang, thank you for the cheer. Oops, I got a little too close and lost track of it. There it is. Okay. It was being covered by shamrocks or something. So this little opening, you can just barely see. We're zoomed in like crazy on the light microscope, but that's what this thing right here is. This is a chrysophyte stomatocyst, and it probably doesn't belong to the one that had that scale that we saw. There's a lot of different species of chrysophytes. But because the scaled organism is the one that people know and put a name on, and this is it's sort of like they go in through a hibernation period. This is their hibernating form. And they create a totally different skeletal structure to hide inside of these things and live through the winter usually. People don't know what stomatocyst necessarily goes with the organism. This is a common problem in paleontology. If you're an organism that makes tracks, sometimes we can figure out what the organism is that's made the tracks, but you don't always know what that organism is. And sometimes it's a challenge, and so there's a whole field called ichnofossils, where they're basically looking at trace fossils, which are like footprints of dinosaurs, footprints of anything. Snails leave a trail when they move around in sediments. It doesn't matter what it is. If it leaves a trail, or it burrows, for example, those things basically, they leave a mark, and we find those things as fossils. But they're not really organisms. They're things that make those shapes. And trying to connect those pieces is challenging. In ichnofossils, in the study of trace fossils, they just give names to the things as though they're species because they see the shape and they know the shape. Paleocastor is this organism that created these spiral burrows, and we found the spiral burrows for a long time, and we never figured out what the organism was. And eventually they found it was some sort of rodent, I think, that was creating these spiral burrows in what they think were probably prairie-like settings. So these are just trace. We'd find the trace, but we didn't know what the organism was. And this is similar to that. It's the the resting stage for the organism, but we don't know what the actual stage of the organism that goes with the resting stage is. And in chrysophytes, I don't study chrysophytes, but in chrysophytes, they just put numbers on these things, which I find hilarious. The first time I heard that, I was like, what? They have a whole book, and they're just like, this one's number 2, this one's number 3. Like, just somebody went through and put a number on each one, and I guess when they find new ones, they just give them new numbers. To me, that's a crazy, crazy way to handle this situation, because remembering, oh, that's number 317, of course. That's harder, in my honest opinion, than learning some crazy Latin name. I mean, we don't usually think of it that way, but putting a crazy Latin name on something actually helps you remember it a lot better than that's number 217 or whatever. I think those are kind of crazy. All right, so I'm going to come back and try to catch this comment. Happy Nightmare says, I'm distracted with my homework from my dinosaur course, but every time I look up or listen, it's like, whoa, amazing. Oh, thank you. Are crisis fights earlier branching than diatoms? I don't know. The chrysophytes, I'll help you with that. It's chrysophyte. I actually don't know the how old they are. They're all stromatophiles. So I think they're probably just sisters in terms of time, but I don't know exactly for sure, and I because I don't study them. I just find them, and I actually don't look at the really old stuff in diatom worlds. Stromatophiles, yeah, like stremens. Yeah, they have some hairs that 3 hairs that basically diatoms have evolved away from. They're a ancestral feature that they lost, a lost character. But the whole group of them have these like little hairs that sets of hairs that come off of them, and I think the chrysophytes still have the hairs, but diatoms don't. So there's some really cool diatoms right around the corner, though. This is again for Claire. There's a Mastigloia. Somebody told me they had a Navicula as their spirit guides. There's some Naviculas in this as well. There's another Celophora right there in the middle. And then there's a ton of stuff in here. It's all, I did a pretty crappy job of making this slide because there's a bunch of junk stacked on top of each other. This would make a bad slide to try to count because this is too dense right here. So you'd get to this and just be like, I can't handle all of the diatoms. I actually might be able to count it, but it's hard to track which things you've counted and which things you haven't counted when it's just in your head. Stromatophile, yeah, no. Okay, what, did I miss something else? Hey, Chippy Flip, how's it going? Sorry, I should give you a shout out. I'm in blabbing mode, I guess. Just trying to keep up with all of the cool questions people have been asking me, and at the same time trying to occasionally at least juggle over to the microscope and take a look at some things. So there's a ton of diatoms here in their field of view. I probably could name most of them, but there's some in there that I don't know what they are because this isn't my sample, it's my wife's. And she probably could name all of them though, if we brought her down and stuck her on the stream. This one, for example, that's Anumastis rostratus. That's an amphora. It looks like Labica maybe. There's another Celophora pupula in the field of view. This thing over here is Navicula oblonga. There's some more chrysophyte stomaticis for you. That's Oligocera ambigua. I don't know. This is a girdle view of a diatom, so I can't tell what it is, but that there is the Celophora pupula. This is like a diatom rave. Yeah, there's a lot going on in this sample. And actually, I was just trying to like look around in the sample, and then Danny brought in this huge crowd of people, and there's been a kind of nonstop set of questions coming my way, and I'm trying to fend them off as best as I can. But I think maybe we could switch to looking at some other material, because I have a whole bunch of slides here, and I just wanted to I'd like to send them all off. I'm going to send these to our friends over at Science Streams. I know that Danny often raids them because I see you people in Science's channel, and I sometimes see Science's people in Danny's channel. And I am sending and have sent Balint some diatom slides, and I wanted to send him a few more. Because he said that they'd look through them and that there was a lot of people that were interested in looking at the diatom samples. So I thought, oh, well, I can send him some more. This is easy to do, and it's for education, so I'm always going to do something if it's for education, and reaches out to his audience and gives them something a little bit more interesting or different to look at than they usually do. Oops, bumped the stage. Alright, this is a sample from, I'm just going to pull this up. Hopefully there's some stuff on it. We're a little close, but this is a marine sample, and I brought this out in part because I know that Danny's famously from San Francisco area, or San Francisco area, someplace over there, and this is actually some material that our friend Pacific Plankton sent me. So, and Pacific was here early. She's one of my moderators, and I'm her moderator. So I just wanted to give her a little shout out. But this is some material that she sent me, and then I'm going to make some slides and send them off to Danny. Question: if you had to defend the honor of diatoms against some foraminifera fans, what reason would you give them to prove that diatoms are cooler than forams? I'm just going to start with, you live in San Francisco Bay. This is basically taken from your backyard. She collects plankton out there off of the Torpedo Wharf, right by the bridge, the Golden Gate Bridge, and sometimes she does little live streams from there. And she sent me this material. I processed it, and then I made some slides, and I sent them back to her, and she shared them with some people. And I thought I could also send some of this stuff on as well to Balint to let them look at some marine diatoms. I don't know these ones to species. I know them to genus, but they taste like Smarties, right? That's all that matters. That's one thing you could say. I don't know if they're going to take that as an argument if you tell them, well, diatoms smell or taste like Smarties, and I mean, what exactly is a foram? It's a snot with a shell. It's basically just an amoeba, like derivative an amoeba with a shell. But if you want to have a contest about it, you can throw this at them. There are 50, I think that's the right answer, all time, 50 species of forams. 50 species. I mean, I have more than 50 species in this slide right here, and they have like 50 total, and that's like all time. I mean, that's terrible in terms of species diversity. And if I was going to have a throwdown with somebody, I'd be like, oh, I learned 50 species. That's the entire group of all forams that there are. Not genera, 50 species. Look it up. I looked at that once and I was like, what? There's 50 species? That's it? I mean, 50? That's a tiny number. And, they make such a big deal out of the 50. I mean, everybody could learn 50 species. That's so easy. I could learn forams in like an afternoon. I've learned 50 diatoms in one day. Okay, how's that? Does that work? I feel like that's a good throwdown. You could use that, right? I think it's true too. I think that's true. I would Google it first and make sure, but I think that's true. It's definitely way less than 40,000 or 1 million or 2 million. There's just not that many of them. So I don't know who you're trying to start a fight with, but if you want to lob an apple of discord into the group, just throw that at them and say, come back to me when you get a real group of organisms, not just like, some tiny little collection of stuff you're making a big deal about. That's obviously just in joke. I love the foraminifera people, and I don't want to start a fight with them, but if they want to start stuff, it's on. I'll do it. Okay, let's zoom in and look at these crazy pores. Biology battle. Let's have a throwdown. Um, sure. This is these little polygonal things that you're seeing are actually this is the frustral of one marine diatom, and these are the areolae, the openings on the actual valve, on the valve face. The tiny openings are actually these things right here, and then these cells are what we call loculate, which means they have an opening at the top, and then a chamber, and this is the chamber, these honeycomb shapes, and then on the inside they have a covering as well with a little opening, and so there's a two-staged, it's like basically a little honeycomb opening on the outside, a honeycomb wall, and then a second layer of silica on the inside of these things, and then all these little details we can see so nicely in the scanning electron microscope. We can get just a hint of what they look like in the light microscope. But if I zoom out really far, you'll see, oh, it's a round thing. We're looking at a round thing up close. So this is Casinodiscus, I believe. Again, these aren't this isn't my group. I look at freshwater diatoms, but I occasionally like to dig around and look at the marine stuff just because it's huge relative to the freshwater stuff. They're very large because oceans have much more wave energy than lakes typically, and it can support larger organisms that are heavier. And so a lot of the marine diatoms that are plankton are much, much bigger than the freshwater plankton. So when we zoom in, you start looking at really small structures rather than what we were looking at, and seeing a lot of really cool detail on the valve face, because everything is big in the marine realm. It's like the Texas of diatoms, where everything is bigger, right? For diatoms, there are some little ones in here. That is a sneaky little freshwater diatom, right, barely visible right there. That's about the size of that little Michiganiana that we were looking at in the last sample, just to give you some context. One of the things that's kind of neat is that all diatoms are single-celled organisms, and that's one species from the freshwater realm, and that is just one organism, where you could probably fit 40 of those things onto the surface of that in terms of size. For the plankton, that is almost always true, and often for the benthic species as well. The marine ones are just bigger. Most of my marine diatomist colleagues, they don't look at stuff at 100x. They just look at it, with 1,000 times magnification. They usually just look at stuff at like 40. They don't need 400. They don't usually need to switch over to the 1,000, 1,000 times. So 400 magnification is usually fine for them to get things even to species. They almost never use immersion oil or have to take it up to the really fine detailed size, because everything is huge. This is actually an argument I often make with people, which is that if you learn the freshwater diatoms and then you find that there's a marine diatom in your sample, you know right away that thing is an alien that does not belong in your landscape. And so if you were to observe it, you would see it. Hey, user plus number, thank you. If you would observe it, you would be like, oh, this isn't a freshwater diatom at all. You can usually tell right away that there is something in there that doesn't belong in there, and that's a nice feature. I usually tell people, like, you could train a high school student to recognize the difference between most marine genera and most freshwater genera. One thing is just they have really big differences in the sizes. There's characteristics that are different, and there are some genera that transect or transgress both marine and freshwater systems. So not all of the genera are exclusively one or the other. Another thing that's kind of cool is we're seeing a little bit of, let me see if I can, how can I zoom out? I don't know if I can get any farther away without changing my objectives. but you see that sort of blue color right there? It's a bit of interference that's being created by the polarizer. This is a differential interference contrast optics on this microscope, which is why the microscope is so expensive. It's actually not the frame or the name or anything else. It's actually this feature. We're using differential interference contrast, which is makes all the objectives more expensive. But you see this sort of bluish color, and then there's sort of like a red color out here. Those aren't real. Diatoms are translucent, or transparent even. They don't have color, especially when the chloroplasts have been dissolved out. But this is what we call physical color. In other words, the light is refracting through the skeleton of the diatom, and its refraction is basically creating colors. And somebody just used, Golganic just used a Freckled Science emote. This is actually one of the things that Freckled Science is studying, I think, for her PhD, is physical color. I think she's looking at other things than diatoms, but diatoms are one of the things that they've identified that have physical color. We know that they're not actually these colors. They're just, it's opal, right? It's clear. So you trust that same high schooler to code your phylogeny? No, I probably wouldn't, but you could pretty quickly learn the differences between things that you can see in the marine realm. Even the small ones look really different. Like this little guy here, he looks pretty different from most of the diatoms that we saw. They have these little hexagonal structures and those loculate valve faces with the cells that have these like weird hexagons on the surface. That doesn't happen in freshwater diatoms. There's not a single true freshwater diatom that's not just like a partial genus from the marine realm that's snuck over that has the loculate-like structure on the valve face. So this sort of weird grid, that's a marine feature. That's just not something we see in freshwater systems. This guy that we're looking at here is actually triangular shaped. We were talking about this the other day on Danny's stream, and I was chiming in about how diatoms, he was talking about an observation is actually something I'd never thought about before, that organisms usually either have bilateral symmetry, like, you know, human beings, tetrapods, everything else, or they have radial symmetry. And I was like, oh, that's cool. Diatoms have both. This is an example of a centric diatom. It has radial symmetry. In other words, it has a central point, and you could then, you have this radiation, this direction, in 3 directions in this case. And the diatom has symmetry that has a mirror plane you could run along the length of it in 1, 2, 3 directions. If it's only 2 directions, this also does have bilateral symmetry, by the way, which means that you could fold it along this axis if it went all the way through. But there's 3 of those. And in true bilateral symmetry, you'd only be able to do that once. Like, it can only fold me one way and get a perfect mirror of myself. In an ideal world, we'd be perfect. But, you know, human faces are not perfect. Our heads are not perfect. But this is what we usually would refer to as radial symmetry, because true bilateral symmetry, you wouldn't be able to do it 3 times. You'd only be able to do it once. But this is an example of a centric diatom, a diatom that has radial symmetry. And all of the round diatoms that we've been looking at, everything that we looked at that was round, circular diatoms today, in all of the other stuff, including this sample, those all have radial symmetry. Anything round has radial symmetry. So there's an example. These all have radial symmetry, right? So you could fold this thing like an infinite number of different directions and get the same shapes. These ones also have radial symmetry. But if I look around a little bit, I'll show you one that has bilateral symmetry. I just have to find one. We're looking at some plankton samples, and the plankton usually are dominated by things with radial symmetry. So here's a Ellerbeckia, for example. That's radial symmetry. This thing does not. That does not have radial symmetry. It's shaped very asymmetrically, but if we were looking at it from the top, and I was trying to be careful, it would be bilaterally symmetric. You'd be able to divide it down the middle. And I'm trying to find a better example of one, because most of the time diatoms just go ahead and announce which one they have, because they have a process that runs through the middle of their body called a raphe that actually, almost all of them actually divides them bilaterally through the diatom. Some of them don't have that structure, but maybe it'll be easier if I switch to the non-marine plankton sample in order to find one that actually has a bilateral symmetry. But that's pretty common. Let's see, what is this? Clear Lake. This is a sample from one of my students that she lent me. She said, "Oh, I have a copy of this one. I can lend it to you." And this slide's fine, so we should be able to take a look at it and find something that has bilateral symmetry, just so I can make my point. Aha, plenty of things here with bilateral symmetry. So let me fix the field of view so you can see stuff, and then I'll highlight that just so we can have that conversation. But I think it's actually pretty interesting, because I hadn't thought about it before as a, why do diatoms have both of those things? And I still don't know. Like why they might have both strategies versus just picking one. So, but you asked a question in chat when we were talking about it. You said, is that happen pretty high up in the diatoms? And the answer is yes. So I tried to answer that, but I think the chat was moving a little too quickly. And but that is the case. It is a major division at the very top of the highest division possible for diatoms is to split the things that basically have radial symmetry from the things that have bilateral symmetry. And then everything after that is potentially things could lose something, like a lost character. They may lose their bilateral symmetry, or they may lose their radial symmetry, but they belong with those things. So this is an example. You could fold this thing. If you were making a snowflake on a piece of paper, you could only fold it along this axis right here through the middle. And if you tried to fold it this way, it would not make a perfect snowflake. In other words, not a mirror plane that you could run through this diatom in any other direction than this direction. It's the only way you could cut it. So clearly bilaterally symmetrical. And I would say that most of the things that are bilaterally symmetrical, I think this is true, are benthic organisms, the things that live attached to a substrate. And most of the things that are plankton generally are radial symmetry. But there's a reason why someone in your chat was asking about why would things have symmetry? Why is it useful? And if you think about, I had a really cool conversation with one of my students once who's a computer modeler. He likes to make 3D models of diatoms. And he was talking about if you're trying to code, like genetically code an organism, the amount of information that you need when you start using symmetry is reduced. If every one of your limbs was a different shape and stuck out in a different angle and had different kind of components to it, then you would need to have, genetic information for every one of those appendages. But when you have bilateral symmetry, I only need the code for half of my body. So my genetic code could actually be reduced in terms of the total amount of information that's needed. I only need to know how to make one arm because I can make an opposite arm that's basically a mirror of the first one. And I only need to know how to make one leg because I can make two. Or if I'm a starfish, I only need to know how to make one structure and then I can just repeat it. And so it's an interesting from a genetic point of view because you can make a complicated organism with less genetic information. I don't know if there's a purpose for that, but you can think about it that way, right? It's very efficient. And most things that have that symmetry, you can actually make more of it with less code because you have the same code, you're just rotating it, right? And organisms know how to generate their skeletal structures through that code. So it's an interesting concept of like trying to be efficient with the genetic information. I don't know that there's like a bigger concept for that. I haven't thought about it enough, and I don't know enough about genetic stuff, but it would be a really interesting conversation to have with like Evil Ozzy, and maybe you, me, and Evil Ozzy on a panel talking about like what could you do with this kind of concept, because from the genetic side, is there really pressure to have efficient DNA code? I mean, I can't imagine that there is, but it feels like maybe it's a byproduct rather than efficiency. Like, one of the things that happens with organisms that have segmented bodies, it's the same way, like worms or anything that has segmented bodies, like insects. If you damage the code genetically and it's just repeating itself, right? Potentially, you could still make that and be survivable. Like, you could add a second set of arms onto a human that came out, from a different place, and that organism probably would survive, and maybe it would be beneficial. So there might be a case for having DNA where the code is replicated, through damage somehow, actually make it beneficial for the organism. So that's a possible explanation, but I don't know enough about the other side of things. I only know my little window, right? So it'd be cool to have a conversation like that. And it would be fun. Yeah, we could do that. Maybe we should talk to Evil Ozzy and see if we can get some sort of a little panel, get some of the science streamers. I know that the Knowledge Foundation did that occasionally. I know that we had a stream that was me and Pacific Plankton and Constababble, and Del Maximum was our moderator. And I thought it was a great conversation. We had so much fun doing it as well. So I think it might be fun to do something like that again. We probably just need to organize it either ourselves or ask the Knowledge Fellowship if they would be interested in hosting it. I think Danny probably has some insight into how we could get that to happen. So it would be fun. It would definitely be a thing I'd be interested in doing. And I think it would be a great educational tool to just sort of express with the viewers how we approach problems like that, and how you could see basically both sides of that. But you need to kind of have a wide understanding of science to make those connections and to sort of sort out what might be valuable. So all of these diatoms that we're seeing are bilaterally symmetrical. I think it's almost all the same species. This is Epithemia. Epithemia are characterized by, I've just been talking about whatever comes to mind tonight, which is totally fine. Epithemia are these guys. In that little picture at the bottom down here, you can actually see that's the one we're looking at. It's that guy right there. It's the same one. Epithemia have a raphe, which is this process on the valve face that the diatoms use to crawl around when they have one. And it runs along the margin on the outside edge, and then it moves towards the middle of the valve and then moves back out to the outside margin, which is a characteristic of Epithemia. And then the other characteristics that separate it from many of the other diatoms in terms of the genus is they have costae, which are like that's just a science way of saying a rib of silica that runs along the length of the inside of the valve, and sort of divides up the inside of the cell wall into these little long chambers. This is sets of costae that are running all along it, giving them sort of zebra stripe structure. And then these little boxy things are the areolae, and the striae are made up of areolae. So for these, you could usually tell which species they are by looking at the number of costae in 10 microns. We would measure, for example, the density of the costae, and we would look at how many striae, or how many of these sets of dots in rows, occur between each of the costi. So for this one, I think you would agree with me, there are usually either 2 or 3 of the little boxy things that go between each of the costi. So this one has 3, this one has 2, this is 2, this has 3 rows, this has 2 rows, 2 rows, this one has 3 rows, 2 rows. There may be an occasional 4 that gets stuck in there, and you can actually use that to help figure out what the species is. Another characteristic for the species identification of epithemia is the shape, the outside shape. Hello? Are you sneaking in? We were looking at some of your samples earlier. I have a slide I made from one of your samples. You know, you have just the...
It's sitting in the hood at school?
No, it's on one of the countertops in there. The other thing that you can use to help you identify it is how far does the raphe go from the margin in towards the center of the diatom. So if it comes about halfway, we get this one. But sometimes these come all the way up. In different species, they might come all the way across, or they might just barely come into the center. So you can use those, the depth at which the raphe sort of reaches into the center of the valve to help you identify them to species. So, Mind of a Snail, hello. How are you? It's been so long since you've streamed. It's been almost a whole month since we saw you last. Does it look like a fancy glass mustache? Well, that's a great question. There's only one way for us to tell, and that's, I suspect, to try to see. So I don't want to do that. I want to just turn it a little bit. Turning it's not very easy to do without a screwdriver, though. What we should do, though, is put a mustache on it. You know, if we put a mustache on a diatom that looks like a mustache, we'll know for sure whether it looks like a mustache, right?
Ty's playing tricks like Tiny around
this
devils.
kind of mustache, or maybe this, yeah, more like this kind of mustache, right?
Mustache!
There we go. Oh, you want the French mustache? Okay, well, we can do that. We'll get rid of that one, and we'll put a little mustache on it, and then we'll see how it goes. And then if you think it looks like a mustache, there's only one way to tell. That's to put an actual mustache on it. And, you know, part of my total joy here is learning to put mustaches on diatoms. Let's see, let's do like maybe 220. No, that's the wrong way. That's like upside down mustache. I don't want that. That one's still upside down. I need it to be like 300. Maybe. Huh, getting closer. 310. It's a 310. There we go. We got a mustache. I don't know that we can zoom out any further, but we got these googly eyes we have to place as well. So let's, do you think it should be, let's see, who asked me for the googly eyes? Mind of a Snail. Where do you think the eyes should be? Like out here on the outside edge, or in here like more normal eyes would be? Like this? I feel like the mustache is appropriately scaled, but maybe it could come down a little bit. You know, like make it a little bit more, take up a little less of the face. What do you think we should do with the eyes, though? Both works for you? As long as it's symmetrical. I'm with you. Exactly. Feeling, that is actually a great observation. It just has to be symmetrical, and then we'll feel okay about it. You like widen everything. Like you like the eyes out here. Out here like that. Like super wide eyes. And then I think probably it needs to be they need to be a little bit bigger. Like maybe like that. Super wide, big eyes. And as long as it's symmetrical, or kind of close to symmetrical, right? Then I think we're doing okay. like this. And then what I want people to do is, it's like a character's head from the Cartoon Network, exactly. And then you should be able to type a googly in the chat, and then you should be able to make its eyes google around. That's just how we roll around. No, googly. Googly. It has to have a Y. Not Google. There you go. There you go. And they'll just keep spinning as much as you want. That's a reward. I don't know how that's rewarding for people other than I find it hilarious. And for some reason other people find it hilarious. We can put googly eyes on anything. Diatoms especially. So, let's see. Where are we? Thank you. Let's see. Thanks for using your channel points for that, of course. You've been online, you've been offline, you've missed everyone. You're back this Sunday for sure. Okay. Well, if you like great music combined with very funny, entertaining puppetry, then Mind of a Snail is the right place to be. They stream on Sunday nights, and only Sunday nights usually. And it's one of my favorite places to hang out on Sunday night. It helps me prepare for the following week. I've had no preparation for the following week for 3 weeks now. So, you know, there's a deficit in my hilarity budget, and so it's important that they're back. When I was on Mind of a Snail's stream, I built this thing, which is, oh, is it even working? Maybe it's not working. Oh no, it's working. Yeah. I built this thing, which is basically me as a puppet, and when I talk, it talks. And I thought this was a lot of fun. And you could just put the mask on, and then I could start talking, and then the mouth of the diatom would move. This is my character Mustachio, who's one of the emotes we have in this channel, and it's definitely the only diatom VTuber that you'll ever find. I don't think any of my colleagues even know what it means when I tell them, oh, I've got a Twitch channel where I put diatoms on and I fill a room full of like 30 or 40 people as we look at diatoms. They don't believe me, for one, and for and for the other, I was talking with one of my colleagues about how I stream not too frequently, but at least once a week, if I can help it, and then and that I have like almost, I guess I have 2,700 or something followers like that. It's not a huge number, but they had a hard time believing that I had that many people that were interested in just sitting around listening to me blab about diatoms. It's a lot more understandable to have somebody tell you that they are interested in sitting around listening to dinosaurs, because we have our bias towards large organisms, and to be honest, they look like dragons and cool creatures, whereas diatoms just look like weird symmetrical things. But and they're tiny and invisible for most people without a microscope. So there might have been amoeba-like VTubers a long time ago. It's not really. So when I say this is VTubing, people get really crazy and go, "That's PNG tubing," because I guess it matters whether you say V or PNG or whatever. The idea is that it's just an avatar. Anyway, you can pop it off. I'll just have to shake really hard, and then basically it pops off my face, and I can go back to talking normal. Goes back in the corner, So it's disorienting to hear my voice coming out of a cartoon character's face. So diatoms look like art to you. Yeah, I agree, and especially to me, because I colorize them and turn them into actual artwork that I do. That's mostly just for fun. I made it because I was doing a collaboration with Mind of a Snail, and I wanted to try it out, and then it was hilarious to me, so I kept it. I just recently bought I bought some of my merchandise from Redbubble channel, just because I wanted to have some stuff, and I bought this Mustachio t-shirt, and when it came, the mustachio was supposed to be in the middle of the t-shirt, but it was like off to one side, like really asymmetric. And I thought, I don't know if I can wear it like that. Like it's not in the center. So I sent them a message and I said, this isn't the way this is supposed to look. And I know because it's my store and that's my artwork on there. And they said, oh, we'll just send you another one, no problem. So they're going to send me another one. And I thought, well, now I've got a shirt. I'm going to have a second shirt. And one of them's going to be in the center and one of them's going to be asymmetric. But I thought I'll just give the asymmetric one away. So if you're interested in having a Mustachio t-shirt, we'll probably do a giveaway for it. And then I'll just let you know in advance. It's a little off center. And if you're a little off center, you probably won't mind at all. So, yeah, somebody changed Claire's purple into green earlier. They changed my algal color, the green one. That was Imanoli. Imanoli, you took your life in your own hands when you did that, because if Claire was still here, she would have been livid. But it turns out when I shook off the mustachio face, it turned back to purple, which is there's a low probability of that happening. There's like a 20% chance of it being purple, and then a second time where it turned purple. So that would be 20% of 20%. So pretty low number, right? Anyway, you want that T-shirt? All right. We'll have some sort of a contest for it. We did a coloring contest before, and I might try to do another coloring contest. And then I usually let my daughter judge the contests. But we might also do just a raffle sort of thing as well. We'll see what we can manage. My stream is very fancy. Which part? The mustache? Because I think we had mustaches the whole time. Pretty early on I started putting mustaches on things. Or do you just mean the bubble? The bubble is fancy. Yes, make you do art. I think that's a great plan. I love the intersection of art and science, and I really like educating people about whatever I feel like educating them about. And I feel like if mustaches and googly eyes are the thing that sells people on it, I'll do it. Especially if it's googly eyes, because we all like googly eyes, right? All right, so let's. I'm going to take this slide off. This one is super boring. It's just got the same diatom on like everywhere, and it's because it was collected from a plant, an aquatic plant, which is where these things usually live, and they are the dominant organism in that environment. And so when the slide was made, it was made from modern samples, and they're not super interesting as a result because they only have one or two different things on it. I don't know what this is. Not if you can imagine it. 6LT6. It's like a mystery. I put slides together for us to watch and see what they were. I'm going to give some of them to science streams, and then I didn't bother to check out what was on them. Oh, mustache. We don't want the mustache out here just floating in outer space. Let's see. I need to do this, and then this, and then I gotta hide these things. Thank you for spending points on the mustache and the eyes for everybody's entertainment. And let me see if I can get this into focus for us and see what we're looking at. Oh, this is actually really cool. This is a project that I have an undergrad working on, and we will probably be using some of the money from the stream to help support this project. It's a really interesting project to me. I have an undergraduate who's working in my lab named Daphne, and I think maybe she hasn't been on any of the streams yet, but she probably will be eventually. And she's working on this project. This is another diatom that just looks like a mustache, I suppose, but bilaterally symmetric mustache shape. This one is the diatom's name is Synedra cyclopum, and we can get a nice close-up look at it if I zoom in. What you can see is One, it has this sort of banana shape to it. Two, it has little striae that we can see in here, and then a section in the middle where there's no striae, or maybe faint lines where striae could have been, and otherwise pretty simple. And what's interesting about this is that this diatom, Synedra cyclopum, only lives on copepods and swimming zooplankton in lakes. It is Synedra. I like your name. Yes, cyclopum. Seems like it should have one eye, but it doesn't have any eyes. It's Synedra, and is the genus, and I think cyclopum's on the web page there. Let me grab it really quickly. So you can, if you feel like maybe I don't know what I'm talking about, I'm not going to be offended. But I'll just verify for you that this is what we're looking at. This is that's the genus page for Synedra cyclopum, so that we're all on the same page, literally. You can see that Synedra cyclopum is looks exactly like what we're looking at here. Banana shaped, a gap in the middle where there's faint striae or no striae, and then you'll actually see somewhere down here under the ecology that it occurs widespread, dispersed in the plankton of lakes, and then it says the report says that it is often attached to crustacea in cool water, and another report that says it's found in lakes of medium electrolyte content where it lives on the backs of copepods. And that is actually the case. This was collected from Gull Lake in California, and this diatom, this banana-shaped diatom, lives attached to the backs of copepods. And if you don't know what a copepod is, hey, Sinophyte, how's it going? If you don't know what a copepod is, I recommend that you go hang out a little bit in Pacific Plankton's channel. She often has live copepods, and you can see them from the marine realm. Pretty common. They're also common in freshwater systems as zooplankton, things that eat diatoms, and fish eat them. But the copepods have these things living attached onto them. And this sample was collected from the plankton of Gull Lake, but the plankton was filled with copepods. When we brought the nets up, basically, it was just all copepods. And we processed it to get to in nitric acid to boil out the skeleton remains of the copepods, and all we had left were just these things, which is kind of cool. So we know that this diatom was basically living attached to the copepods, because we have some pictures of the carapaces of the copepods with the diatom still attached to them in place. And then when we had a sample that was full of copepods, we had a sample that was also full of Synedra cyclopum. And what this means for me as a paleoecologist is, if I'm looking through my samples from this lake and I find a bunch of Synedra cyclopum, that means that we historically would have had more copepods likely present in the water column during the time where we find these. And the advantage of that is that diatoms fossilize, but copepods do not. And typically we can't tell the concentration of copepods. So this actually is a way that we are planning to reconstruct the history of copepod abundance in Gull Lake by looking at a core that we collect from the lake and then counting the abundance of these things, with the connection that the more of those that we find, then likely the more copepods were present in the lake. And so it provides a way for us to basically, I sound a little congested. It's because I am just a little congested, but I'm okay with sounding congested. The Studio Cornix is here, one of my favorite streamers, and she also connects science and art together from the other side. I'm sort of a scientist who does artsy things, and she's sort of an artist that does sciencey things, and so we're like a perfect pair in my mind. And Pacific Plankton is also the same way. She's an artist who does sciencey things. So I really make nice combinations with those kinds of people because I can help them with the science side of things, and they can help me by being artists and showcasing all of that cool stuff that I dabble in, but don't have exactly the types of skills that they do. So so the value of these is basically we're going to try to reconstruct. My undergrad in my lab, who's been in there for 1 year, she can recognize this diatom, and that's the only one she needs to know. And we can look through the samples, and we just can say diatom or synhedrocyclochrome. Like, just counting the total number is easy. This is an example of something that anyone could potentially do. You don't need to be a specialist to be able to recognize this thing. It's very distinct. It's banana shaped, and it has these weird little stripes on it. You are unlikely to confuse it with anything else. And so I'm going to train her in identifying these things, and then we're just going to give it a run. No one has ever tried to do this before, by the way. People know that this thing lives on copepods, but most diatomists don't care about the concentration of copepods in lake systems. They are mostly concerned about what are the diatoms telling us, not what are copepods telling us. And what I'm interested in is it tells us something about the linkage between the food webs. And so there's a really interesting aspect of it for me, which is that that we can use the diatom as a record of a thing that we cannot observe directly, which is the copepod populations. And the copepod populations are probably linked directly to fish populations, which we could observe, but it's difficult because their fossils are much larger than we typically have material for. And so this is actually a really interesting concept for me, which is to apply something. People know something about the ecology of this organism, but they're more focused on other things, and so they don't really care that much about it. And I mentioned this to one of my other paleoecology colleagues, and he was like, "Ooh, that's cool. That's a really cool idea." And he was very supportive of it, so I thought, well, we'll give it a try. But we'll probably be using the funding from Twitch almost exclusively, unless we get a grant. I wrote a little grant to try to cover it, and I don't know if it got funded. But if it didn't, we're just going to exclusively use Twitch funding, basically, to pay for this. And then I'm almost certain that it's going to come out with some sort of very interesting results. Nobody's tried to do it before. And then we'll almost certainly try to publish it. So we'll be able to get something that basically we developed more or less just by chatting about it, and a project that we didn't intend when we started to do this project. We had no concept of what we might do. Hey, how's it going, Froza? Sorry, I didn't see you come in. I was busy blabbing. But this is something that happens in science, which is basically a lot of times you don't prepare for everything you might observe. Hey, thank you for the subscription, Gerbo Potato. That's super cool. You don't always know what it is that you're going to get. It's a bit of a surprise. Some of science is exploration. And so we'll be basically making some connections, but like I don't have, like I have funding for the things that we designed the grant to pay for, which was not this part of the project. And so we're going to basically be using some of the Twitch funding to fund Daphne's research, and she'll be working on this project, getting paid to analyze these samples, and then if I can help it, she'll either be the first or the second author on the paper, depending on how much she feels about writing about the topic. And then my other graduate student, Laura, who has also already been partially funded by our Twitch stuff to do stuff in the scanning electron microscope, will also be probably an author on that paper. So really cool, connecting the research of some of my graduate students with some of my undergraduate students, giving them some really cool scientific research experience, and being able to fund it mostly by just blabbing at people about things I just already like talking about anyway. So a pretty cool system. So especially to people like Gerber Potato and Paleologizing and Chippy Flip just now dumped another 5 subs on the channel. This is the sort of thing that helps us fund that research. I mean, honestly, it's nice to be able to do it and not have to scramble around and try to find like how are we gonna get this project funded. It's a pretty cool project. It's gonna probably result some very interesting aspects and will lead to the student presenting paper at maybe the next GSA or something like that. So keep your eye out for it if you're going to any of those national meetings. Frozen, what did you get for your spirit guide? I didn't see it. I'm hoping it was a Sinedra. Oh, Denticula. All right, there you go. All time from now forward, you will have Denticula guiding you on your path. Look at all the people who just got subs too. Very cool. Disillusioned and Broken Symmetry are people that are always hanging out in my channel anyway, so they're going to be very excited to find out that they got subscriptions. So, Chippy Flip, great job. Hey, it's Crossover Carissa. I was hanging out in Crossover's stream right before I came here. I was listening to her sing. She's one of very cool streamers on Twitch. I spend a lot of my time hanging out on Twitch in the community. I like to hang out in science channels because I'm a scientist, but I really, really like to hang out in the music channels, and I think most of the music streamers have a pretty good handle on the fact that I'm there. They're like, who's this weird person who's like a scientist who's hanging out in my music channel? I'm very, very much excited about music all the time, and I just think it's brilliant that people have an audience. You know, like I was a musician. I was in bands when I was in college and a little bit in grad school, and I mean, we played out at bars to crowds of like 30 people. We were happy with those kinds of numbers. And when I see streamers on Twitch and I think, wow, they're here just like with this massive crowd of people that are checking out their singing, that you can just come from anywhere. You have such a nice audience to be able to pull in, but you're giving them a free concert. You know, like a concert that they can go to just every other day or something. It's extremely cool. So what did I play? I was a bass guitarist. And when I was in high school, I did a lot of singing. So I really appreciate your singing. I was in a small town when I grew up, and I was expected to do a bunch of plays because there weren't a lot of people who could sing. And I don't know that I sang that well, but my the person who was in charge of all the musical aspects of plays would constantly tell me if I just had some music lessons, I probably could be a great singer. She loved my voice. It's raspy today, but she would always just tell me like the quality, the tone of my voice was just amazing, and I was like, okay, whatever. I wasn't planning on becoming a singer, but I don't have quite the ear for singing a tune very well, so I probably do need lessons if I wanted to do it. But you're right, there's so much good music on Twitch, and I almost always like to, if I don't come out of my channel streaming and raid another musician, either going after an artist, a musician, or a friend of ours who's doing science or art or music. That's basically all I do. Almost never go into a gaming channel, but I almost always end up in either the art streams or the music streams because I love the community and I want to be able to share music with people. I added this thing very recently to my channel, which I'm calling Metronome with a G, which is this. It just randomly blurts out. You gotta go. Yeah, I know how it is when you get to the end. When you get to the, it has like a little timer, and then I have it randomly select for musicians that I regularly listen to on Twitch. It's this thing, and it just picks one of the people that I follow that's a musician, and it recommends them. And Carissa, you're in that list. So just randomly, my stream will blurt out, hey, why don't you go give Carissa a follow? and I really love that you brought your people in here. So I'm super happy that you came through and get some. I know how it is. You get to the end of the stream, you just want to like go relax. So I'll try to come back and take a look at what was going on. Just a second here. And try to read a little bit. Is it kind of like funding science with science education? A little bit. It's incidental funding. I mean, I can write grants. I often try to write grants to fund stuff, but there's not a lot of money for science. If you're not in the medical profession, there's a lot more money in medicine. But for pure science, for looking at something that's like diatom stuff, it's very competitive and difficult to bring in money. So, like getting any money that I can donate. I mean, the thing is, I'm not going to use it. I don't need somebody to spend $5 on me. I have a salary. I do a job. I get paid to do my job. I don't really want the money to come to me. And I was going to stream anyway, so I sort of feel it's like an easy way for me to give back to my students who are in my lab, and it seemed like a better cause than just giving it to some random organization. So did you talk about the content of said project? Yeah, we've been talking a little bit about it just now, recently, but we haven't fully developed it, so we'll see. I do have a small grant that might fund some of this as well for this particular project, but it's going to be a fun one, and I'm very interested in seeing what happens. So Science Nerd, what's going on? Yeah. Thanks again for the raid, Carissa. And I will formally give you a little shout out here. Crossover Carissa. And if you don't know who Crossover Carissa is, and you're wondering, like, do I want to go listen to some random music channel? She's a vocalist. She plays mostly keyboards and does loops, and she's got a spectacular singing voice. And she does requests. She's super friendly. The community there is great. I wouldn't recommend a streamer for music if I didn't think that they had a great community. And also, Unmagical Me, thank you for coming in with the raid. Unmagical Me knows what's up. He's in a lot of, or he or she's in a lot of the music streams I'm in. Have a good sense of taste. Your favorite is Denticula creticola, because it looks like a microscopic submarine. All right. Well, if you looked around in the denticula, let's see. So many of my friends on Twitch are music streamers, and sometimes I do a little bit of woodworking and try to make people things, instruments for streamer friends of mine. So, you played drums for 15 years, Froza. Nice. I haven't played bass in a long time. I have sort of like arthritic-y hands now. I'm old, so yeah. Singing stream coming. Well, I thought maybe Hannah Rebecca Music was going to come visit me while they were in Wisconsin. She threatened to, and we had plans, and then she just decided she wasn't going to do any streaming for a while, so we kind of missed it. But I thought I was going to have to sing if that happened, so, but I also thought I would have to practice a little bit. We'll see. Very, very cool. All right. If you love music streams, so that was me. Yeah, it was Calvin Thomas. It was just an example. I also feel like I'm participating in scientific research. Yeah, exactly. That's part of the cool part of what's going on. So, oh, did it just come up? Oh, that was mine. Yeah, okay. It would have been a lot of fun. We could have had dueling kalimbas. Why not fundraiser special music stream? Like invite music streamers and do a multi-stream stuff. That would be really fun. I don't know if I could convince that many streamers music streamers to do something, to do a fundraiser. But I like organizing people. At some point I would really wanted to do like a 24-hour microscope stream collection, where it would start with like Dell Maximum and then come to me, and then I would give it to Pack, and then we'd give it to like whoever. We'd end up with like Spider ID and Studio Cornix, and like all the we just hand it as a raid train from one to the next. I think that would be a lot of fun. Let's see. Diatoms do have special xanthophylls specific to them. Yes, Thiazoxanthin, yes. We should do that. We had, when we first started on Twitch, when I first started on Twitch, there was like 3 people who had used microscopes to stream onto Twitch, and it was like a tiny world who's the person who got me into streaming on Twitch. Just basically putting a microscope and showing water bears and some rotifers and things. OpenSet, who still streams, but very rarely anymore. And a tiny world hasn't streamed in, I think, a year at least. But she was amazing and got me interested in doing this. She talked me into putting my SEM on Twitch, and then Pacific Plankton, who started just a little before me, who I was watching and also thought, okay, I can do this, because I don't. I know it seems like I'm pretty calm talking in front of people because it's my job, but the idea of like putting my face up and constantly talking in front of people was not appealing to me at first, and it's still not all that appealing to me. But I realized that having my face showing is probably more appealing to people than just only the microscope stuff. But there wasn't that many of us, right? Talking to people is kind of challenging. And being able to talk, I don't know, I've been streaming for 2 hours and 30 minutes almost, and I haven't shut up. So talking to myself for 2.5 hours while I'm answering questions in a room with just me and a microscope, it's intimidating to think about doing that. So, but I think it's actually made me a little bit better at communicating with the general public, which is important for me. As a scientist, I think it's actually really critically important to be able to talk to the general public, and I think that's another thing why Danny brought in a whole bunch of people earlier. If you're not following Paleontologizing, you should be. And he's been hanging out here. I think he eventually signed off, but maybe he's lurking in the background. But he brought a whole bunch of people over and then just hung out and chatted with me about really cool stuff, which is nice. It's nice to be able to like just have a dialogue about interesting things with scientists sometimes on Twitch as well. As much as I also just like talking to average people, I think it's kind of cool to have dialogue in front of people so they can kind of see scientific thought and process. So, pass the baton. It's a fun way to do it, to do like a raid train. Yeah. When I socialize, I have to stay silent and alone, recharge my batteries. That's actually me. I spend most of my time with my headphones on. I don't want anybody to talk to me. I just want to be like quiet and doing my work. And I think that that it's a challenge to be around people for me. It's it doesn't seem like it is, but it's not something that I crave. It's more the other way around. I'm definitely an introverted type personality, and like in big rooms full of people, I'm usually try to find a corner and hide. So I didn't mean average in terms of your abilities on Magical Me, but if you want to take it away, you can. You're an extrovert. Systemic isolation has been hell. Yeah, I can see how that would be. When the pandemic first started, I feel like it's ended for us, and I'm gonna hit that for a second. I know it hasn't, but I feel like it's ended where I live. So when it first started, the neighbors were like, "Oh, how are you guys putting up with the fact that you don't have anybody around and whatever else?" I was like, "Oh no, this is good. I don't mind this at all. Like, I could live in my house with just my microscope and my music and my kid and my wife, and I don't need the rest of you." It was totally fine for me. I had no problem living in total isolation like that. I would make a great hermit, like if I could just find some quiet wood space where I could also have internet and a microscope and an SEM. But it didn't bother me at all. I know there's a lot of people it had a really negative effect for. Today, here in the county that I live in, I think 3 or 4 days ago, they said after 780 some days in a row, we have no cases of hospitalized people for COVID in our county anymore. and our COVID numbers in the county are generally hovering around 0, with occasionally 1 or 2 cases being reported like once a week. None of our universities dropped the mask policy. Everything is basically back to normal here. So like we are essentially past it at this point. I don't know if it'll recur. I mean, if it does, I think we're all prepared for that as a possibility. But here in Indiana, in this little part of the world, we are nobody comes here apparently. But I feel like we're past it. You don't. I mean, if there's 1 or 2 cases in the county being reported every couple of days, I feel like I don't need to wear a mask anymore. I'm vaccinated. The concentration of the viral load is so small at that point that there's almost no chance that I could get infected. I would have to be exposed to somebody who was definitely positive for a fairly long time, probably to because I'm triple vaccinated or whatever. It would be challenging to basically, I'd have to work at trying to get infected where I am right now. And as we just talked about, I'm kind of an introvert, so like I teach my lecture and then I go back and hide in my office or one of my labs. I don't socialize with people very much. So, but I'm thankful about that. And I know that's not the case everywhere in the U.S., but that is the case here. Yeah, and we have a pretty small county, so and I'm in a rural place for the most part. So, and I don't mean to minimize the effects of it. I'm still aware that there's a pandemic going on, but I feel like I'm Taiwan or something where basically there's been almost no cases. Because we're down to that point where it's not really a big deal. And when I hear somebody cough, I usually go, oh, that person has a cold. If it had been a year ago and somebody coughed, I'd have been running for the other side of the room, right? And we have all of these COVID kits that for testing that my wife has been, like when she sees them become available, she goes and buys some of them just because. And every time she gets something like a sinus infection or a cold or something, she takes a test and it's like it always comes out negative. And I'm just like, yeah, I don't think we're in a place where you could really get COVID right now. So I'm happy about that. It's a break for me to not feel like I'm in a pandemic and constantly in fear of illness. But I definitely know that it's not that way everywhere. So you're in a region that has lots of cases, but you still feel a bit hopeful that it will fully end by the fall. I don't know. It seems like it's definitely declining in most places. Pandemic isolation has you burnt out. Lab hermit life. Yeah, I had to blast music and make things more lively during the last semester. When it first started, I was doing a lot of, I was doing like, Zoom kind of hangouts and just to kind of see people and chat with them a little bit. And I hadn't seen my family in a while, which, like I don't care about talking to my neighbors so much, but not getting to see my family is usually a bit of a challenge. So, but they're all, they're all vaccinated and double vaccinated and triple vaccinated or whatever by this point. So there's not a lot of threat for them either. I just have one more slide I want to look through before I decide which one of these I want to send off to science. I think we've looked through all of the other ones, and I just wanted to take a quick look at it. Sorry about the blinding light. It wasn't my intention. Just trying to see if there's good things in here. Earlier in the stream we were talking about other things that we find in the samples. Let me fix this. I need to, we were talking about other things that we find in the samples that aren't diatoms, and the samples are digested in nitric acid or hydrogen peroxide and nitric acid. And so most of the things that are organic get dissolved or oxidized in that process. And it mostly leaves behind things like silica, but sometimes pollen is really resistant. Anyway, we talked a little bit about chrysophytes earlier in the stream, and these are all chrysophytes. These little spiny things here, they look like globes. They really look more like Christmas tree ornaments, as we'll see in the scanning electron microscope. These are apertures or openings, and you'll always find an aperture or an opening in a chrysophyte stomatocyst. You might not be able to see it if it's on the other side of the sphere, but they're spherical shaped and they have a little opening. There's this one, here's the one on this one, there's the one on this one. That'll let you very easily tell that you're looking at a chrysophyte stomatocyst versus something else. And one of the things that you can see in this sample is that there are a lot of chrysophyte stomatocysts. Here's a whole bunch more. And we're still at 40x, so I'm going to 400 times magnification. So I'm going to, real quick, throw it into oil immersion. This juggling two things at once, so that we can see those things a little bit more closely and clearly. But there's a whole little cluster of different chrysophyte stomatocysts, and I need to do this thing again because brightness doesn't know how to adjust itself without it. But you can see all of those little spherical things have openings in them. Here's the opening, here's an opening, here's an opening, here's an opening. Sometimes they have little spines and things around them. But these are fixed samples, so we're able to look at them with oil immersion very easily. And this is 1,000 times magnification with my camera sort of stacked on top of it with doing a little bit of additional magnification. They're not a crystal formation, they're an organism. They are a type of golden algae, and looks like a dumbbell. There are some phytoliths that look exactly like dumbbells. It's actually just two spherical things in that case. They look a little dumbbell shaped, but this one is a chrysophyte. It's a little asymmetric, like a plum. And then these ones are also all chrysophytes. So this sample just has tons of chrysophytes, more chrysophytes than diatoms, I think, which is fine. Here's some more chrysophytes. There's a diatom that is Pinularia borealis. This is a stack of, I don't know what that is. Pseudostarcira brevistriata, I think. There is a Cymbopleura. And these are pieces of pine pollen right here. This yellowy colored stuff that you're seeing is that's a shredded piece of pine pollen. Those are some chrysophyte cysts in them. That's a Pinularia piece. That's a Cerarella piece. Just a fragment. There's another chrysophyte. That one's got all kinds of dynamic spiny things sticking off of it. That is a Celophora levissima. This is a Cymbella. I don't know what the species for that is. It looks like maybe it's Cymbiformis. This is a piece of Onidium. This thing over here is a little tiny Gomphanema. There's a bunch of little chrysophytes in there. That's that little Gomphanema again right there. Tiny, tiny little Gomphanema. That leaf-shaped thing. Cerarellas. Some more chrysophyte cysts. That's an Oligosyra cluster right there, a colony. That is, that's Oligosyra valida. There's an Epithemia gibba, just a piece of one. One of the things is that this is a sample that I've my materials, so I can very quickly tell you what the species are for most of the things in here because I counted it. I counted it like 15 years ago, but I still counted it, and most of the things I still know their names. That is a piece of a sponge spicule. So sponges put nasty little toothpick-shaped spines inside their body made out of silica. You can tell it's a sponge spicule because it has this structure, which is called an axial canal. It runs down the middle of it. Sometimes people confuse sponges with diatoms because diatoms can be long, toothpick-shaped or banana-shaped things, but a diatom will never have a axial canal running down the middle of the silica. And then of course these are solid silica. You can see that here that there's no holes or openings. And that blue color that you're seeing, or purpley color you're seeing right in there, is just a refraction of the light. So, do they generally stay in a colony? The diatoms that live in a colony, the Alicocera that live in a colony, they have specialized spines that they use to hold on to the neighboring siblings in the colony. That's a nice Epithemia gibba right there. Sometimes you can see those Epithemia gibba in their living states on Studio Cornex's streams. Studio's had some housing issues that's made her very angry and left her unable to stream for most of the week. But hopefully they've been resolved. Yesterday we had all kinds of drama. Apparently the SWAT were in her neighborhood, swatting some people. This little thing right here is a Pseudostarocera pumila, I think is the species. You're angry. That dude went to jail. Okay, good, because he was. It seemed like he was being defiant with the authorities. Never a good plan. Always, when the SWAT team shows up, you should just let them take you, because they're gonna take you eventually. I don't know what you think's gonna happen, but you're not getting out of that one. There's some little interesting chrysophyte cysts. There's a lot of really weird ones. This is one, and this is another stomatocyst. This one has ridges, like spiraling ridges that are going around the outside of it. It's not super clear from this field of view that that's what's going on, but through optical dissection I can tell that it has a bunch of spiraling ridges. Right here is the aperture, and then the spirals are all coming around the valve like this, and then this one just has like a reticulated surface, which you can see right here very easily. A little bit more clearly visible in the microscope itself, though. Doing some work and lurking. Did I just hear they tried swatting Studio? No, not Studio. Her neighbor. He was holding his girlfriend hostage. Oh, he sounds like a charming person. It sounds like maybe he deserves to go to a little quiet place where he can think about what he's done. Holding his girlfriend hostage. I think you probably mean his ex-girlfriend. I'm just gonna guess. I'm just gonna guess that step one in girlfriend maneuvers is don't hold them hostage. This is Pinularia, again, borealis. Pinularia borealis is an aerophytic diatom. It sounds like he sounds like a great guy. I don't know why he's got to hold his girlfriend hostage. He seems like he's very charming. Ooh, that's a big diatom. It's a giant old Cerorella, just taking up most of the field of view right there. I think it's Cerorella, not Iconella, but I think that's a Cerorella now. Still, there's a chrysophyte inside the Cerorella, and there's a little diatom. That's also a diatom. This whole thing is a diatom. And if we had one of those marine diatoms in here, we would just be sitting on top of one of those and show you how huge they can be. Where did the sample come from? The sample is from a bog in the San Juan Mountains, up in the high elevation area in a place called Cumbers Bog, or Cumbres Bog. I don't know how it's pronounced. And it was the subject of a paper that we published in 2012, and it's in my research portfolio down there somewhere. If you click on the research button under the About Me thing, you should be able to find it and download it. I think the lead author, I'm not the lead author, I'm just a component of it, but I do talk about the diatom record a little in it. And this is from the past when the bog was actually a lake, which is part of my interpretation. This is Calanese. It has this triungulate shape, so undulation, 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 and it looks like a circus peanut, or double E circus, double peanut. And then this is Calanese. I believe it's Salicula, but I could be wrong about that. It could be Schumania. It's one of those two probably, though. And I counted these while I was counting my dissertation as extra work, because apparently counting 2,000 diatom slides wasn't enough for me. And I needed to have at least another 60 or 70 slides that had nothing to do with my dissertation that I could count at the same time, because that's what I was doing. This one's got these really cool little wicked spines coming off the back side of it. There's the, I think that's the aperture over here, and those wicked little spines are like sticking off the bottom of it. I also counted another 500 slides during my dissertation from Flathead Lake and the river systems around Flathead from modern collections, on top of the 2,000 slides that I counted. And I left a hole when I graduated that no one can ever fill. My PhD advisor is just like, you've wrecked everything for everyone who's ever come through here, because no one has ever counted 2,000 diatom slides from a single site before in lake system history, and I did that for my PhD, and then another 500 plus another 100 from 2 unrelated projects. So everybody who comes through there after me is very mad at the fact that I did all of that, because they're like, I just want to count like a normal amount, but I really like diatoms. That's all I can say about that. I wasn't trying to break records or anything. It just happened. These little cistula things here, I think that's Cimbella cistula. These are the stigmoids, stigmoids, and they are on the ventral side, on the belly, instead of on the back. We often look at, on this channel, we look at Afrocimbella, which have its stigma on the dorsal side. Cimbella always on the ventral side. I just got carried away. I just liked what I was doing. What's wrong with that? Do you use any vision tools like ImageJ? That's a good question. Chippy Flip. When I was a graduate student, we did use ImageJ to measure things. When we first learned diatoms, we used ImageJ as our primary tool for that, because we didn't have access to high-powered computer programs. But this is a Leica, the DM2500, and I have software in my lab that will allow us to measure the same way, that came with the computer, but is integrated, so I don't have to do any thinking. I can just draw a scale bar and it will tell me how big that is, and it's calibrated for every one of the different objectives, and it will also measure things like area, and you can count features so that you don't have to track it in your brain. So if you were trying to, for example, count how many of the areolae are in this row, you can go put like click, 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 and it will count and then tell you how many you counted. But ImageJ is the tool that we started with, and we would use it regularly, and so I learned how to do stuff on ImageJ, and then quickly decided that I liked the I don't know how much money they spend on. You have to spend like $4,000 or $5,000 for the software that comes with the microscope. It's really expensive because they got you, over a barrel with it. But I decided I liked it a lot better, and since we already had bought it, I was just going to use that instead of going back to ImageJ. On my home computer here, the one that we're looking at stuff with this microscope that's here from, I'm borrowing it from my lab, but my wife is mostly counting on it on the desktop behind me, so I just bring it over here. Hey Shafard, how's it going? How are you doing? Hopefully that rest of that anniversary stream you were doing turned out pretty well. The South Park game you were playing made me laugh a lot. We don't have it hooked up to any of the softwares or computers. I just have it hooked up to the camera that you can see up here mounted on the top of the microscope. And the images are calibrated after we take them. We have a stage micrometer, so we can take a picture of it and then take a picture of our samples, and we can just use the pixel number counts to measure it if we need to. More typically, what I would do is if it was something I wanted to take real pictures for publication or for statistical analysis, I would take it back to the lab and do them there. And the pictures that we collect with this one are mostly for building a reference guide. If she wanted to, if she wants to take some pictures so she has them, we have a camera mounted on it, and then once you have the microscope set up and the camera set up the same way, the pictures are all the same magnification because it's just pixels, right? It's not 1.5 times, it's 1.5 times 1,000 times, so that's actually 1,500 times magnification. So do not be deceived by the mere 1.5. That's my camera's magnification of the image that you're seeing. So I can zoom out, and now we're just looking at 1,000 times magnification. This is a fun little diplanese right here. I like being zoomed in because the zoom actually is a lot clearer on this camera when you're using it on a microscope, and the image looks a lot cleaner for people at home. There's not as much diffraction as I get closer, and the optical zoom on my camera has better magnification and better resolution than the actual camera that came with the microscope by a lot. So as I zoom in, we don't actually get any loss of resolution because it can't really happen to us. So this is a 20 megapixel camera, and you need something like 5 megapixels at 1,000 times magnification to to see things clearly. So no matter what I do, I can't really reach the magnification that we need, the limit of it. Oh, there's another giant Sorella, and then it's sitting right next to this little epithemia right there. Oops, you can't see the epithemia because we're zoomed in too far. Got those eyeballs going, Chef. It was really funny just hanging out in your stream. I'd never seen that game before, but just watching you break stuff is hilarious. So, Keonce makes some pretty cool stitching tools for making images of huge areas, yes. There's a tool that people use, I can't remember the name of it, but they basically have microscopes that are set up to take, and you can call me Jeffrey, that's fine. I mean, that's my name. Game was it right? Yeah. I can't remember the name of that software, but they have these microscopes that are set up to, they'll just take pictures and move automatically across the slide, and they'll just keep taking pictures, and then it stitches the whole thing together, and it has to do it. The image is Z stacked, and then it's moved, and then Z stacked, and then moved, and then Z stacked, and then moved, so that everything is in same focal height. So all of the images basically, they won't look normal. The diatoms actually won't have the normal depth of focus. So like I can tell the shape of things by using optical dissection. You wouldn't be able to do it with those images. So things that had a hump basically would look kind of flattened, but or they would to me. They might not. They would look abnormally flat from the way that I'm used to looking at them. But the tool then would allow researchers to go through and circle all the diatoms of the same species, for example, and it's a really neat system because you can collaborate with other people. You can all look at this thing that's in shared space and then decide which things you think belong to the same species. And I wish I had one of those microscopes that could scan and take one of those pictures and stitch it together. I don't have that kind of money, and neither does my university. And while I think it would be really, really cool to do, it's like an excessive instrument. Like we can do that as humans. We don't necessarily need to have that kind of a tool to do that. but it is super cool. And I think one of my friends was involved with it. It's in some sites where they're specializing in doing this sort of stuff in Germany or Austria. And my friend Hannah, who's German, a native German, was working with them and promoting it and talking with people about how they could use it, and was showcasing some of the stuff that she and I were collaborating on to showcase it to me. So yeah, this is a nice, a cool epithemia. This is Epithemia sorex, I believe. It's either sorex. No, I'm pretty sure that's sorex. Let's see. This is Epithemia. You didn't have to look it up. Oh, I need to spell things correctly. Epithemia. There we go. You didn't need to look up the fact that it was epithemia, or you didn't need to look up the fact that it's sorex. Because if you knew it was sorex, that's pretty good. That's in the realm of yeah. This is sorex. I'm pretty sure. So we'll bring that back to the Twitch channel here. Didn't have to look up epithemia. Still an accomplishment, but it's good that you can identify them. No idea of the species. Well, this one looks like Captain Crunch hat, so that's how I usually remember it. Sorex is the Captain Crunch hat. Let's double check that ID though. Oh no, I think it's right. This one actually just slightly different shaped. Let me see what else we have for epithemia. I would start with sorex basically as my first guess. And
eyes playing tricks.
Oh,
like
it's gonna get
tiny
googly
round
eyes, huh?
devils.
We could do that again. The more I stream, the more it's a mustache hat. You can't unsee it. The googly eyes. Water buffalo horns, yeah. This is one of those that needs to have the eyes way out on the outside edges, like out here. Now it looks like it's a trap. The Star Wars character, right? And then you should be able to Captain Crunch hat. Yeah, that's the way I usually think of it. And you should just be able to type googly, and then the eyes will spin around, and you can Google them as much as you like. They're the hammerhead diatom, exactly. Let's see, we could put a mustache on it. I think that would really probably help. What about this mustache? Oh, that's the normal one. We did that one earlier. What about this bushy mustache? Yeah, a good bushy mustache. Well, I do have a question though, which is, which way does the mustache go? I'm a little confused. I feel like this is the way the mustache goes, but I guess I could... Admiral Akbar, exactly. The wall-eyed look. What to do with this mustache, though? Let's see. Let's get it straight. I do like the crooked look on the mustache, but maybe not quite so crooked. No, we need 55, it looks like. Do you think we should do a poll? Do you think it should be like a little mustache right here? I kind of like that look. Or should it be a big mustache? You know, like this. No. Okay, opposed to that. The other question I have is, what about... I'm trusting Studio to give us the right answers, because she's our artist in the channel. But what about this? No? Because I'm confused which way the mustache goes. You're the artist, yeah. But could the mustache go this way? Because look at that. You don't think so? But what about it? No? Because I feel like it could. That is a smiley Look at the smile. Look at the smile. I feel like it could. You're opposed to that. Okay. Well, I'm aware. That's totally fine. I feel like we can trust you. We will have to trust you with the right answer. Oh, wait, it was 55. Look, exactly. Look at how happy he is. Although he looks pretty happy this way as well, to be honest. And I really do think he looked better with the little tiny mustache. You know, just like here or here. Here? Here. I feel like it goes there. Somewhere in that range. Tiny. The tinier the better. He looks funnier the tinier I make it. It's like he needs. I needed to use the one with the monocle. He needs a monocle is what he really needs, because I think that is classy. He looks very classy. Perfect. Okay, good. Makes him look like Sid the Sloth from Ice Age. It does. It very much does, Mooch. Good observation. Okay. I think this is a great place to stop. I feel like we've had a lot of fun. I've looked through some slides. We've had a great conversation dealing with both science and fun stuff. And I've managed to run my voice, even though I've had this really stuffy sounding vocal fry all night. Still managed to make it work. The question is who to attack. I have choices, and my choices are science ones, science streams, who these slides are probably going to go to. Glorgana, who is doing some After Dark Monarch Caterpillar artwork. And that's it. We got those two choices. Oh, and Freckled Science. Those are my primary recommendations. So if anybody has a strong preference, I'm willing to listen. Any of those would be fine. For where to send people. And then we'll call it an evening. A very fun evening. It's like 12:30 here, and I have to get up early in the morning and take my daughter to school, like a responsible parent. So, Diatom's Attack is live. Well, I don't know that guy very well. I don't follow him. Let's see. Where to go? Where to go? Caterpillar art? Butterflies? Oh, is this butterflies in a microscope? And microscopy. I think maybe we should go visit Freckled Science. I feel like that's a good choice. She's doing butterflies in a microscopy. She's in our wheelhouse, and we should have science streams, I think, on my channel next week. So we have good overlap with all of them, but I haven't rated Freckled Science in a while. So why don't we give our friend Freckled Science the raid? Feel free to attack her like crazy. And she's doing microscope stuff with butterflies. It sounds perfect. And she's on the West Coast, so she's going to be there a little bit longer than probably anybody else, and still active for a while. So I want to thank a bunch of people. We've had a really great stream with a lot of action. We got googly eyes from Chippy Flip and a mustache. We had community gift subs out of Chippy Flip. He gave 5 of them. We had a raid from our friend Crossover Carissa. She brought in 7 viewers. A very great musician. You should definitely give her a listen. We had a subscription from our new friend Gerbo Potato. Thank you very much, Gerbo, as well. You and Chippy Flip are heroes for the evening. A follow from Mooch, who just gave us his first comment. We had a bunch of people just using the rewards in the channel, which is cool. User plus number cheered 100 bits. As I mentioned, very much appreciative of any money people bring into the channel that we use for student research. and Crowfang and Claw also 100 bits. We got a subscription from Alt Miss Frizzle that's subscribed as well, a teacher who's probably going to use some of her stuff for content. We had a huge raid from Paleontologizing early on, and I want to thank all those people. So I'll start with that, and we'll catch you next time.